Okay, I'll let the stragglers at the back make their way forward. But welcome to everybody to this very exciting 20-year anniversary symposium of Generation Scotland. My name's Cathie Sudlow. I'm the director of the Usher Institute and head of the School of Population Health Sciences here in Edinburgh, and lucky enough to be the current director of Generation Scotland, the wonderful resource that we're here to celebrate and find out more about today. So there are, I think, over 350 people registered to attend this event either in person or online. You come from all around Scotland, across the UK, and in some cases further afield. Please feel very, very welcome, including to those of you who are joining us online. In particular, we have almost 50 participants, Generation Scotland participants, who've registered to be with us in the room today, and a whole load more who are joining us online. So you are particularly welcome. This endeavour, this resource would not be happening without your generosity of spirit and your time and everything that you have done to help guide the resource through the last 20 years. So a welcome, one and all. Throughout the event, we're going to invite you to engage with us using a gizmo, to use a technical term called WooClap, it's not one I've come across before, but there are a number of other options are available. And you can join via the QR code that's on your flyer. If you want to, you can use your mobile device. You don't have to. If you do participate, it's kind of voting and questionnaires and so on, survey stuff. There's no personal data collected about you at all. So everyone's privacy and security, as with the resource itself, is respected. This event is being live streamed and recorded, so for anyone who hasn't been able to join us today, there will be an opportunity to catch up via a video link. In terms of quick housekeeping announcement, there aren't any planned fire alarms today, so if you do hear an alarm, it says here, please evacuate calmly. Okay, I'll let you interpret what that means. And the fire exits are at the back of the room and down and to the right. Okay, over that way. Is that behind me, Fiona? Okay, there's one over there as well. So just to make sure you know where those are. There are some toilets on this floor just at the back there, but also on the ground floor, including an accessible toilet on the ground floor, and there's a lift available for anyone who needs it. Now, I've been asked to make some comments about this clock here, which I can't see now, but which is reading a sort of peculiar time. So it's both upside down, I think I've worked out, but it also goes backwards. So please, unless you have kind of the ability to make mirror images and inverted images in your brain, please don't rely on it if you need to run off and catch a train or a bus. Use your mobile phone or your watch or nudge your neighbour for help. We have some coffee breaks and lunch today, so we've got some breaks about 11:10 and this afternoon at about 2:10. Before each of those breaks, a trolley is going to proceed up the aisle with coffee and tea on it. So please stay seated just for a brief time at the start of each break to allow the trolley to get to where it needs to so that you can get what you need off the trolley, which will be served at the back of the room. Lunch is going to be at just after 12:30. We'll be taking some photographs. Just for those taking photographs, please make sure that you don't photograph anyone wearing a yellow lanyard. That signifies that they've elected not to be photographed. So please, could that be respected? If anyone needs help of any sort, we have plenty of Generation Scotland team members. This is not their full-time role. They're actually all scientists. They manage data. They administer the resource, and they do all sorts of other things. But today they are here to assist you, so please do find them. They're all wearing blue staff badges if you need any help. We'll finish today at about 4:00, and after that there's going to be a drinks reception. So if you signed up to attend that, do please join us for drinks. Now the real business starts. So it gives me great pleasure to welcome to open today's proceedings Dame Anna Dominiczak. Dame Anna is, importantly, one of the original gang of four illustrious scientists, now 20 years older than they were when the whole thing started, but they don't look a day different, not to me anyway. So Anna was one of the original gang of 4 who set the Generation Scotland resource up. You'll hear about the others, I'm sure, during the course of the day, and you'll hear from them as well. But Anna is also Regis Chair of Medicine at the University of Glasgow and currently the Scottish Government's Chief Scientist for Health. I can't think of anyone better to open today's proceedings, so Dame Anna, please, you're very welcome.
Right, that's it. Thank you very much, Cathie. She's very modest. She runs this show currently, and we need to be very grateful to her and her team that we're here today. So maybe that's the first thing I should say. I have many hats that she already mentioned, and they showed on the right top side by these logos. But I think today I am here because for the last few years I've been a chair of the Scientific Steering Committee, and that allows me quarterly to hear lots of good things that are happening with Generation Scotland. And I'll just share very quickly a few things, and thanks to the team for slides they've made for me. And I think it shows incredible longevity of this study. So this is the summary. More than 40,000 participants. In fact, the last number, that more than 16,000, makes it comfortably more than 40,000. And that makes it a very, very large, useful longitudinal cohort, and particularly NextGenScot, i.e. adding young people that Cathie started and others followed, is really, really great. And you'll see some data coming from this. And as you look at the resources as a researcher, you want to do another study tomorrow because it's so tempting, all the data gathered. So just very quickly, what do we have? So that's this initial recruitment, 2006 to 11. Gang of 4, I think, met first time about 2003-04. David, we'll hear more about it from David later. But the real recruitment, 2006 to 11. And you see these resources. So there is genomics, there is epigenomics, there is CHI, the Community Health Index linkage, and there is this enormous percentage of participants who agreed to be recontacted. If you look at other similar studies or generally population studies around the world, around the UK, that percentage of recontact, and you see on the next slide, 100% agreement to recontact is unprecedented from what I have seen. You see, of course, smaller numbers because of price for it, proteomics, metabolomics, and brain imaging. So fantastic, what we would call high-fidelity phenotyping. And of course, this resource can serve many, many studies, and we're going to see it in a minute. That's the more recent recruitment. Look at the dates, 2022, 26, 22. We were just coming out of COVID. That's an enormous effort that more recruitment has been done through these not so easy times. And you see again, more questionnaires, special characterization of those younger participants, 12, 15. And what I already mentioned, consent to be recontacted 100%. That's huge. I think we need to thank colleagues who delivered this profusely. Now, that's full cohort with lots of data sources, and as I said, you would like to get your team to start investigating it now for yet another question, yet another study. You see the common questionnaire data. This is very deep, and very, very extensive. Now, who is accessing this and why? Some great stuff is going on. You see that when genotype data have been released, a large peak, that line, There will be another such peak, we believe, in 25, where the next gen genotyping will be released. But I think the interesting things here are that the light blue that you see behind me, these are external investigators who come and ask for data to study, actually many times exceed the internal investigators. And that means that this study is now known nationally and internationally and a real resource for asking difficult questions in medicine, public health, etc. I'm also hugely impressed by this publication's topic summary. The Chief Scientist Office has 26 clinical research and specialty groups, 26, and I thought that was plenty. But here there is more than 26 topics, medical and around medicine, ethics, law, etc., using the data. And you can see how that increased over the years. If you look at 2006, 10, and now 21 to 25, almost all parts of medicine used the Generation Scotland data to answer questions. And I think that's really great accolade to everybody involved, the participants, the people who discuss each approach to access data ethically and appropriately, etc. And our board looks at this regularly. Now, there's been lots of great publications. Apologies if your favorite one is not on this list. Maybe I should add that yesterday there was fantastic publication from the Gang of Four. I'm not sure, he'll tell us later whether GS data not used, but Nature on Pain, which is one of the topics that Generation Scotland is also addressing. But you see here what we would have predicted: COVID, cancer, cardiovascular disease, aging, and of course cognition and mental health, where a lot of work over last many years has been focused. I would like to show you now a photograph from 20 years ago that I found in my collection. If you recognize yourself on it, put your hands up, please, please, pretty please. I see you, I see you. Yes, any more? Yeah, not many. If you're not on it, as Cathie already said, there'll be opportunity to get photographed in the next break. So when in this room colleagues will meet in 20 years, you'll be on this photograph. So please don't shy away, come and be photographed. And on a more serious note, I also found the questions that we posed to Generation Scotland early work applications for funding, including Chief Scientist Office at the time and Scottish Funding Council. And these were research questions in 2006. And I think as you hear our speakers, you'll see that majority of them, not all of them, but majority of them have been at least partially or completely answered. Heart disease, mental health, bone disease, and adverse drug reactions. All these things have been addressed at some point by fantastic data gathered by Generation Scotland. So that's all I wanted to say. And now we come to the real Generation Scotland interesting stuff, because our next speaker is David Porteous. Professor David Porteous is a geneticist. His bio that he gave us is very modest, the most modest bio I have seen of any professor in many years, but I know better than what he's written there. So David has done fantastic genetic studies over the years, but Generation Scotland has been his baby from the beginning, fighting for money, fighting to keep it going, not to let it go, to make it bigger and better. That's been David. So yes, there was a gang of four, but David kept us sort of on our toes throughout. So David, please come and tell us about the history and the real soul of Generation Scotland, please.
Thank you very much indeed, Anna, for being here for the whole day, which is fantastic. And as we'll see, there are a number of really important people who are going to be here for the full day as well. I am allegedly fully retired and have been for several years, but I don't think I can properly retire from Generation Scotland because I can get to watch all the stuff that's new and happening, and a lot of it building on where we started off all those years ago. And I think my task today is to talk to you a little bit about that early history. What was the idea behind it in the first instance? How did we work it up into a viable project? And some of the pitfalls and problems along the way, because we shouldn't ignore those either. So, the real proposal here was that we had a problem. We were witnessing this rise and rise of chronic disease in our population, partly to do with an aging population, but also to changing environment, changing behavior. And the proposal was that if we took account of that problem and we matched it with what was a genuine revolution in genetics, maybe we could find out the kind of answers to these big problems that we're so passionate about. So the idea was a Scottish family-based study. We were going to make life difficult for ourselves. We wanted to collect information about families, not just individuals. We wanted to ask questions about genes and environment. And we wanted to look not just at physical health, which was the predominant issue of the day back 20 years ago, but also mental health, which was at that time really underexplored. And we wanted to be able to do it from birth to old age. Now, if you started at birth, you'd have to wait a long, long time before you were able to look at problems that different individuals were facing in terms of their health. But was there a way that we could look back in time and capture early life events and use that to try and understand what was happening in later life? There's also this classic issue of nature versus nurture. And it should never have a V in it. It's always about nature and nurture. How do we get the full product of the influence of biological factors inherited over the generations with environmental factors that impact us on our daily lives, whether it's about where we live, our occupation, our education, or profound unexpected events, about which more later. The big question, of course, is which genes matter and which environments matter. And they're going to matter differently for different people. And we wanted to try and tease that apart. We know that we are going to, almost inevitably within a family, witness devastating bad news for one reason or another, whether it's cancer, cardiovascular disease, diabetes, you name it, it's going to happen. The big question really is, well, why one family but not another? Why one individual and not another? And that's when it really matters to be able to measure both the biological aspects, which are written into your DNA, and the environmental aspects, which are how you've been brought up, where you live, what you do, what you're exposed to. So that's why I'm going to take you back here to this one slide about the Human Genome Project. When we started out, not one genome had been sequenced. And we had a 50-year gap between the original description of the model of DNA and generating the first consensus map of the human genome. That 3 billion letter discovery of the human genome cost $3 billion to do, and it involved thousands upon thousands of investigators throughout the world to put that together. The beauty of this is that once you have a consensus genome sequence, every new sequence can be paired to it, and you look for differences. The differences are the differences between our biological risk factors. Move on 10 years beyond that, we now had tens of thousands of individual genomes, and the cost had plummeted. It was only $3,000 to do a whole genome. Move on to today, it's less than $300. We're talking about a price to sequence a whole genome that is less than the average consultation with your GP or your consultant in a hospital. So this is incredibly valuable information that we have now. And it was possible to anticipate that well before it happened. It was a certainty. We knew we could rely on this. The other question, though, is about if you're going to do a family study and you want to look at the environment, why would you do it in Scotland? What are the particular advantages of doing so in Scotland? Number one, top quality health research. Our medical schools were preeminent in the world globally, tackling all sorts of different conditions, as Anna has pointed out. But more important in some ways than that was that we had these long-standing, high-quality health, social and demographic records looked after for us by the NHS in Scotland. And you've already seen a snapshot of this, but these records go way back. So from the 1970s, 80s, 90s, increasingly, long-standing records were collected, archived, and available to do epidemiology, to ask why are some of the conditions evolving at the rate that they are, and where are they most prevalent, and what factors are influencing them. On top of those, comprehensive records about hospital visits, maternity records, prescribing data, mental health, dental and cancer registry. We also have the Scottish Index of Multiple Deprivation, which tells you why your health is going to be affected by where you live, education and occupation, and births, deaths, and marriages. So there was a potential that was almost unique in the world at the time to be able to do this retrospective study and add to it the genetics revolution. Oh, now. Wait a minute. Oh, I've missed one. Yeah, so this is the now infamous or famous Gang of Four. No one person, no one institution was ever going to do this in Scotland. But we are very fortunate that we are all, irrespective of the medical school, within 2 hours traveling distance of each other. And I was here in Edinburgh as the genetics expert, as you've heard. Anna, preeminent clinical investigator in Glasgow. Andrew Morris at the time in Dundee, though now in Edinburgh. And Blair Smith, the wee boy, the youngster, was at the University of Aberdeen. But each of us brought something different to the table. Clinical expertise, Andrew Morris, the pioneer of using medical records, electronic records. Blair Smith, a leading light in public health and in pain, and continues to the day now in Dundee. So it was the 4 of us coming together, sharing our experience, bringing our colleagues along with us, and knocking on the door of the Scottish Government that really was the tipping point in making this work. Before we could start, though, there was a lot of groundwork to do. There was the consultation with all of our academic colleagues. Science and medicine is a competitive business. There are some people that say, no, I don't want to get involved in that. You'll be taking away the one thing I've been working all my life on. But we broke that down. We could show that if we just pulled together, we would all benefit. Everyone would have a chance to be able to dip into the data and do their own thing. Public consultation, absolutely vital. It's wonderful that we've got so many people who are volunteers for Generation Scotland here today, because without that, we would be going nowhere. Lots of public consultation on what people expected, what they thought we were already doing, why we weren't doing more, and how we would protect their personal information and make sure that it was to the common benefit. And then, of course, before you do any medical research, there is a huge raft of regulatory review and ethics review, quite rightly and properly. that it is vital, if we're going to do this, that all of the ethical issues are mapped out, and that we could be seen as trustworthy. So we had very strong public interest, a clear public benefit. We put privacy and transparency right at the top, and we ensured that all the data was going to be kept safe and secure. And it's very gratifying to us in Generation Scotland that we have had feedback on that we are a model for others to follow. So, roughly speaking, this is a very simplistic breakdown of it. On the right, we've got the NHS Scotland doing all the blood tests, doing all the prescribing, and keeping their data safe and secure. And on the left-hand side, we have Generation Scotland doing the same. The bit in the middle is key, though. The data was going into a so-called data safe haven, so that neither the NHS could see our data, nor could we see their data, because it had all been anonymized. That meant we could keep it safe and secure for every participant, but we were able to nevertheless get all of that data out and analyze it in a way which was sound and transparent. So when we started, we had this broad consent, ethical consent, to do genetic studies, linkage to records, and to both academic and commercial studies. Important point, the NHS does not make drugs. They rely on companies, biotech companies, to make the drugs. They then assess them, then they decide whether they can afford to prescribe them. Very important point. So we wanted to ensure that we could engage with industry, biotech industry, pharmaceutical industry, etc., to push the research forward. The key features, which Anna again has summarized, are that we undertook multiple tests and measures at the point of recruitment, matched up to samples of blood and urine, and then we did comprehensive genetic analysis linked to the electronic health records, with follow-up through record linkage and recontact, remembering that we could go back in time and also forward, so creating de facto a longitudinal study from day one. It was not all plain sailing. We got generous funding from the Scottish Government to get kick-started and for a continuance of that funding, but that ended in 2013. Fortunately, Andrew McIntosh, you'll hear from later, who is our real golden star in the area of mental health, wanted to set up a big study. He won money from the Wellcome Trust, and he embedded his study in Generation Scotland. So indirectly, we could keep going and expand our efforts. And then in 2019, I won another big grant from the Wellcome Trust for NextGenScot, which you've heard and you'll hear a lot more about in the future. And we assembled a completely new team. Most of the people that are in that original team are here, but everything evolves. And by early March, we were all set to recruit new volunteers, except this happened. That put a pause on all of the kind of research that we wanted to do. For good reason, the NHS had their clear priorities. We wanted to see what we could do to help. And you'll hear later from my colleague from Bristol, who will talk again about this. But basically, COVID-19 changed everything. We had to stay at home, protect the NHS, save lives. So we had to ask the question, what about the indirect health effects on health and well-being? How are people coping and feeling under lockdown? And how could Generation Scotland help? And again, here, the big advantage was we had data from before, during, and after COVID, so we could ask those big questions. 18,000 responses from our adults, for whom we had measures both before and after the pandemic. One of the key findings was that at a level never previously witnessed, life satisfaction plummeted. We'd been through, if we compared our data post the impact of COVID to the impact, for example, of the banking collapse in 2008, and the consequences, that had a tiny effect on life satisfaction. COVID had a massive effect. Anxiety and depression increased, unsurprisingly, but it was important to be able to measure those things. We partnered up with colleagues in Glasgow to ask younger people how they were getting on. Teen COVID life. We had 5,000 people responding to our questionnaire that we were able to send out. One of the key observations, and you might ask more questions about that, but you'll get more answers later in the day. Increased feelings of loneliness. Loneliness is not about being alone. It's much more complex than that. It has a much bigger impact. And of course, we also were able to get a very early impression of just how impactful COVID was in terms of education and well-being. And we're seeing perhaps the end of the tail in terms of GCSE results coming through. But it's long-lasting and it's been of significant impact. So I think that the key messages I have here is that this is a wonderful study, though I say so myself, but it's one that is absolutely dependent on collaboration. Collaboration across competitive academic institutes that are normally fighting for every penny of grant money that they can get. They came together, they pulled together, and it was due to the Gang of Four working hard in each of their institutions that actually came off. Teamwork. We wouldn't be here today if it wasn't for the current team or the previous teams and the original team that we put together to start Generation Scotland. Partnerships also beyond Generation Scotland, of whom there really are many, and it would be wonderful to mention them all, but we've collaborated all across the globe. It's been always fantastic to be able to sign up another partner for that academic endeavour. Transparency of purpose. If the message isn't clear, you're not going to get the support. The fact that we get so many people willing to take part in questionnaires after they've enrolled is a measure of the fact that they obviously are seeing something valuable. And hopefully we will always be able to do that, and that transparency of purpose is the way forward. Undoubtedly, the quantity and quality of health data that we were able to tap into and create ourselves has been vital to its success. But absolutely not least our volunteers. Every single volunteer, every single volunteering family that stuck with us and agrees to yet another questionnaire, it really does help immeasurably, and we would have nothing without them. So I'm going to end just by thanking each and every volunteer for their gift of time, personal information, sample donations, and trust. It's an invaluable gift to Scotland and the wider world. It's a rich and powerful legacy, and I think it's got a bright and vital future. And with that, I will stop, and thank you very much.
Thank you, David. There will be opportunity for discussion at the end of this session. We'll have 15 minutes, so get your questions ready. And our next speaker is Professor Riccardo Marioni, who is a professor of molecular epidemiology and aging at the University of Edinburgh. He is a co-investigator of Generation Scotland, and you'll hear about biomarkers. And he's done some really, really interesting stuff on genomics and epigenomics, and I'm sure he'll tell us more about it. Please.
Thank you so much, Anna, and good morning, everyone. So I'm going to give a very brief overview, hopefully fairly lay in terms of some of the biomarkers that we've measured in Generation Scotland, so covering genetics, epigenetics, and proteomics, and some of those applications in research from my own group. So in a nutshell, what we're interested in, or one of the things we're interested in the group, is taking biosamples, whether that's saliva, blood, or urine, asking what can we measure from those samples, And then once we measure things, can we find certain patterns in those data that help us better understand who's at risk of going on to develop a disease, and kind of how often or how far in the future can we predict who's at highest risk. So that could be cardiovascular disease, dementia, cancer, mental health outcomes, so on and so forth. So something we've already heard about from David is DNA. So here we have a cartoon of the double helix, and I kind of think of this, if you untwist this, you would end up with a ladder with 3 billion rungs on it. So that we get a ladder with 3 billion rungs from our mother, 3 billion rungs from our father, and that kind of long ladder can also be split into chromosomes, so kind of 22 autosomes, and then the sex chromosomes as well. And at the majority of the rungs on this ladder, we look at exactly the same. However, where we differ in terms of the rungs is kind of what dictates features like hair color, eye color, and certain genetic predisposition to disease as well. So DNA is the same in every cell in our body, whether you're looking at the liver, kidney, brain, or any tissue. And as I mentioned, what we're interested in is looking at these, where the rungs differ, and do those differences associate with health outcomes? So a study led by Mark this year, he's taking photos, so you'll probably see him around during the breaks and ask him questions about this. He asked, are there any rungs which associate with major depressive disorder or depression? And he found almost 700 points on the DNA ladder where there are differences between those who have depression and those who have not been diagnosed with depression. And interestingly, some of these rungs are possibly indicative of opportunities for drug repurposing. And also interestingly, whilst we do differ in terms of our genetic sequence based on ancestry, what Mark found was that there was some similarity in the patterns across different ancestries. So the rungs that differ with depression are common across different people from different backgrounds. But whilst DNA is fixed in every cell in our body, there are chemical additions to DNA called DNA methylation. This is an epigenetic mark, and these are different in each cell of our body. So, for example, if you have, like me, 2 brain cells, hopefully, those 2 brain cells would have different methylation patterns in them. However, the pattern in those 2 brain cells would be more similar than if you took a liver cell from me, but they would all be subtly different. And the purpose of methylation is kind of to act as a bit of a dimmer switch. So if you think, we got our genetic sequence, we have genes, and those genes need to be turned up or down during life. So whether that's during adolescence, menopause, anything you can think of, or if you have a cold, certain genes need to be upregulated, others downregulated. So these chemical additions can come and go, and they can help turn genes up and down. So in the cartoon we have here, hopefully you can see it at the back, I've now got 4 brain cells, and we can see that in 3 of those brain cells, on one rung on the DNA ladder, there's this this methyl group attached, and on the 4th brain cell, there's no methyl group attached. So we would say at this particular rung on the DNA ladder, there's 75% methylation. And we called the methylation sites CPGs, but you don't need to worry about that. But then this is actually data from Generation Scotland and from around 5,000 people in the cohort. And here we're looking at one particular rung on the DNA ladder, and we're asking across these 5,000 people whose ages you can see on the x-axis here, so ranging from 18 to 99. Then on the y-axis we have the percentage of methylation at this particular rung on the DNA ladder. And for volunteer 1, we can see that they have around about 75% methylation at this particular rung. The blue line here is representing the average trend for the 5,000 individuals. So you can see that volunteer 1 is aged just over 25, but if we were to try and map that to the average person with 75% methylation, they're probably going to be about 80 years old. So this individual at this site Their methylation resembles that more of an 80-year-old than the average 25-year-old. And there's been a real explosion in research into this over the last decade or so, which GS has been at the forefront of this work, asking why? Why do some individuals seem to be aging faster than others? Does this actually correlate with lifestyle factors, smoking, drinking, obesity? Is it predictive of future risk of disease or mortality? And this is kind of a concept we think of as biological aging. So whilst chronologically we all age at the same rate, are some of us aging faster in ourselves than others? So a couple of years ago, we averaged information not just from one rung on the DNA ladder, but from thousands of rungs, and we came up with a formula that would be predictive of chronological age. So this is work by Elena Bernabeu, a former postdoc in the group, and what Elena found was that when we applied this formula into a set of individuals outside of Generation Scotland, we get a correlation with age close to 1, but most interestingly, we get a median error of 1.7 years. So what that means that if we took samples from everyone here today, that for half of you, so let's say those sitting on the left-hand side, we'd be able to predict your chronological age to within 1.7 years of your actual age. And I think for about 80%, we'd be to within about 4 years prediction accuracy. And again, this just ends up with generating more questions. So we can see there's an individual, you won't be able to see in the mouse, but does this work? Oh, very, very faint. But there's a point up there where the chronological age is, again, around about 30, but the predicted age, based on the methylation patterns, looks to be over 60. Again, why is this happening? And we can link this to the electronic health records that David spoke about. So we can see, do the people with the oldest looking biological ages go on to develop certain diseases over time? Methylation is also fantastic at, so these chemical changes respond to different lifestyle factors. One of the strongest ones is smoking. So this is a study by Ola Kubowska, who's a PhD student in the group. And basically, we can look at the methylation on your DNA, and we can tell with almost perfect accuracy who's a current smoker, who's a never smoker, and who's a former smoker. So the lines here, basically, if we have a value of 0.5, that would be a coin toss as to guess who's a current smoker, or if you're a current smoker or a never smoker. Perfect accuracy is 1, and you can see that we're at 0.98, so 98% for discriminating current smokers from never smokers. So this is all good and fun, but why is it actually useful? So when we answer the questionnaires to Generation Scotland, or for the volunteers here, I did the questionnaires myself for the new cohort, we maybe estimate things slightly wrongly at times. So for example, how much alcohol do you consume? How much do you smoke? Well, it's difficult to remember how much, well, yeah, sorry. How much you've had to drink, but in the sense of, I had a glass of wine at home, how big was the glass, what strength was the glass, how many units was that, I don't know. But you can guesstimate roughly. But methylation gives us a slightly more objective way to assess these things. So we know smoking, drinking, cardiometabolic health, these are major risk factors for cardiovascular disease, cancer, dementia. So can we try and augment what would be on a pen and paper questionnaire by using these biological samples to help improve risk prediction? Another good example with smoking would be 2 people smoke a cigarette. One person might take a couple of puffs, then throw the cigarette away. The other might be inhaling and smoking right down to the filter. But on a questionnaire, you would honestly answer, I smoke 1 cigarette. But it's quite a different exposure in terms of the biological impact of that cigarette. So some other work we've done in Generation Scotland, we've looked at things like blood pressure, alcohol consumption, BMI, and other waist-hip ratio and body fat. And methylation gives us a really powerful tool to explore the impact of lifestyle on our health. We've also looked at it directly to predict things like type 2 diabetes and other health outcomes, showing that it has the potential to improve and to augment current clinical risk predictors. So I mentioned methylation is a bit of a dimmer switch to genes, and then as genes get turned up or get turned down, we create more or less protein. So this is kind of the end point of production. And Generation Scotland also has a fantastic data set of proteomics. So we have around 500 proteins measured in 20,000 individuals. And this is some more work from Ola. It's not published yet, so getting a sneak peek at some of the results. She split the cohort into individuals aged between 40 and 70 at the time of joining in 2006. And she asked, do any of these 400 or so proteins associate with a hospitalization due to cardiovascular disease over the 17 years of follow-up? And you can see the number of cases that we have on the right-hand side here. So over 600 having a composite cardiovascular disease event, ranging from a couple of hundred with heart attack to 100 with stroke, and 250 who had a death due to a cardiovascular outcome. And in this analysis, Ola found 48 proteins to be associated with a future risk of cardiovascular disease, of which some have been reported in the literature before, but 24 of these were novel, so potentially providing new insights into the risk and the pathways of cardiovascular disease. However, all of this work has been done in blood samples, which can be quite invasive. Many people tend not to enjoy giving a blood sample. So in Next Generation Scotland, which we've heard about a little bit already, some of you may already have donated samples using these kits. We had an online sign-up process. Heather will talk about this later. And then we sent a spit kit through the post, asking you to spit down the funnel into the tube and then post back the saliva kit to the lab, where we will analyze that for genetics and epigenetics, so the methylation. And to date, we got around about 11,000 samples back in the lab, which is absolutely fantastic. And this is the largest saliva methylation resource in the world. And one thing that we're now going to do before the end of the grant is to try and recontact everyone to ask, would you please be so kind as to provide another sample for us? Because it's interesting to look at a single snapshot in time for your health, but actually how do things change over time? And even say from last year, from 5 years ago, whenever it was that you donated a sample, have things changed? So just to illustrate how things can change, here's a graphic illustration of aging from approximately age 18 to, it looks like a mugshot in the bottom right, but I think early 30s. So things like hair loss of sight, or having to wear glasses, these are kind of fairly gradual processes. But then when you extend that to health things, does diabetes come overnight? Does cardiovascular disease? Does dementia? No, these things take time to develop, and the power of longitudinal data, so having serial samples of saliva, we can ask questions. Do certain patterns change linearly over time? Or in terms of risk signatures, do you see a massive spike just before a diagnosis? Or, yeah, all these sorts of things that we can explore with the data. So it's an incredible resource, and I think as we build this new biobank, it provides opportunities to really explore change in these risk signatures over time. So if you've changed in diet or weight or started smoking, stopped smoking, how does that influence your biological health? And with that, I will stop and just thank everyone in Generation Scotland, particularly the volunteers. Thank you ever so much for everything you do for the cohort. And also thank you to my group. And yeah, thank you.
Right, we have 15 minutes for discussion. We ask David to come to the hot seat. Do we have mics and questions? Yeah? Okay. Organizers, should I stay here or join them there? Stay here. Yeah, because the mics. Okay. Right, so brief questions from the audience, and when everybody is thinking and delivering the questions by machines or verbally, could I start asking David, clearly we've been seeing a lot of work recently, particularly in cardiovascular diabetes, but also cancer, other chronic diseases, about polygenic risk scores. Are polygenic risk scores a help to your question you posed there regarding risk and prediction? Should we rely more on polygenic risk scores? What do you think?
I think the polygenic risk score, which is summing up the effect of every single gene that you've measured, is a possible way forward that could have big impact. I think actually from our own work, the example that Riccardo mentioned of the work that Mark Adams and Andrew McIntosh, who I know has now joined us, is probably the best example from our work. I think what they've done is take it beyond simply the risk score. What they've done is to ask, how does that risk score relate in terms of the function of genes that are associated with it? So that...
Are we not on?
Oh,
right.
I'm apparently now on, but maybe people at the back could still hear me because I do project. So I think that example of Mark's study that's been published in arguably the most prestigious journal that we ever have a chance to publish in, called Cell, is the best example that we have because it ties together all elements of what we're about, which is to use genetic variation, sum it all up, then ask how does that relate to biological properties within the genome, which genes, and then how those relate to proteins that the clinician wants to know about, and how those might go up and down in relation to an illness. But I think what Riccardo's done is to show just how open the field now is, because we've got this bank of proteomic information and DNA methylation, which I think really is the big future going forward. So Riccardo, carry on from me.
I don't think there's much to add there, David. I think you've kind of said it all. I think it's, polygenic scores are lovely in the sense that they are fixed, they are static. So your polygenic score, your genetic risk for depression is the same at birth as it is at age 50 or age 70. Yeah. Whereas the other elements, the proteins and the methylation, they're constantly changing. So with the genetics, it's easier to know. Not on. Is that better? Oh, yeah.
Yeah.
Sorry. In terms of chicken and egg, we know that the genetics is the start, nothing precedes that, whereas is the methylation or proteomics, are they cause or consequence? So that's much harder to disentangle when it comes to prediction. Okay, so could I try to look into this polygenic risk scores sort of future? I think what's going to happen, and it might happen actually later this week, where 35,000 cardiometabolic physicians are going to meet in Madrid at famous ESC. I think there will be a debate to add, we use clinically in guidelines, in clinical practice, so-called score 2, that is in cardiovascular disease predicting 10-year risk, and our treatment, giving statins, lowering blood pressure, etc., is dependent on this. There is now a number of fantastic papers published, adding polygenic risk score on top of score 2. Actually, simple equation, because they not related for reasons that you explained. Polygenic risk score, we are born with other things, other risks we acquire because of variety of things that happen during lifetime. So I think the two together should now be part of clinical practice. And I am going to speak for this in Madrid
on Friday, and I'll come back and tell you what we got. So I think we're going to change clinical practice, at least in cardiometabolic disorders. Okay, other questions from the audience? Is there anything on the machine? Yeah? Machine.
So there's one online, and anybody who's joining us online can submit their questions on the WooClap. And it's, are autoimmune diseases included in investigations?
Great. Right. David.
The breadth of autoimmune diseases is wide, and it's very important. But if we talk about issues like the role of inflammation in those autoimmune diseases, this is actually, I think, one of the really big issues that this type of work may be able to address in ways that hasn't been possible before. I think there are two things that are perhaps worth thinking about. With the autoimmune diseases, it tends to be something that waxes and wanes. It tends to be something that is triggered by some event and shows itself quite strongly, and then hopefully it may settle down. Others are, in the broader sense of them, can be very much ongoing and life-changing. I would say that it is actually one of the biggest opportunities that has not been possible yet to tackle well. I do think that this is a way forward. I'm just going to put in another pitch for an area that's not tackled as well as it should. I think we're doing a really good job in mental health now compared to 20 years ago. The other deficit, women's health. I'm afraid the sad story is that men are more interested in their well-being than they are of their partners, which is a tragedy. But there's some work that we've done on, for instance, on the menopause, which I think has really been important and has contributed something important in the field in general. I think it deserves much, much more attention. And I'm hoping that that will also come through in the work that we do in the future. Again, using these tools, I think we're able to be much more broad in our ambitions. I hope that happens.
Okay, and we need microphone for Cathie. Right?
Thank you. So, David, you spoke very eloquently about how important it was to put the effort in to recruit people in families, so people who were related to each other. Often what we have seen subsequently in large international studies that have sought to disentangle the genetic and environmental influences on disease is that people who are related to each other tend to be discarded from those studies and seen as a bit of an inconvenience. Do you think there are some missed opportunities to actually really exploit that relatedness in Generation Scotland and other similar resources, and what might those be?
Thank you. I love that question.
Was this planted?
No. But I should say I'm a member of Generation Scotland. I'm also a member of the UK Biobank. And what Cathie is partly alluding to is the fact that the UK Biobank, for practical and strategic reasons, went for as many recruits as possible. And the simplest way to do that is just to ask as many people in the population to sign up and ignore any issues around relationships between one member of the cohort and another. Nic Timpson's in the audience. I'm a great fan of ULSPAC. You'll hear more about that, and he's leading Population Research UK, about which we need to listen as well. As a geneticist, I just found it unbelievable that anyone would want to set out on an endeavor like this and not know the relationship between the participants. The relationship between the participants helps you partition the indirect and direct effects of genetics. The thing that's really neat, though, is the bigger you get, the easier you can see undisclosed relationships. Because the UK Biobank is so big, you can actually look at the relatedness of every individual to every other individual in the cohort. So I'm still a big fan of going for families. I realize it's much more difficult, but in a way, that's another strength of doing things in Scotland, recruiting locally. There's a wonderful study, not unlike ALSPAC, but just looking at families. I'm a member also of the Aberdeen Children of the 1950s. That's where I was born and brought up. That study, by being centered in one town, and which enrolled all babies born in the 1950s, has been fantastic at looking at social demographic change and its impact on health and well-being. So I think there's room for everything, but if you're going to do genetics, please, for goodness sake, ask questions about relatives.
Great. Any other questions? We have 5 minutes. We can have one question. I see the hand up there. And I would like to finish with something on aging and methylation. Because, go. Question, please.
Hello, thanks so much. Can you just say something about the data you're holding? The quantities must be absolutely astronomical. I mean, can you give us a feel about how many bytes per data subject you would have, and how is this actually stored? I mean, what servers are being used? Are you contributing to the energy crisis?
Riccardo, you go.
Yes, and sorry. So the methylation data, I want to say is it in the region of 64 gig? 114 gigabytes for the... So it's not enormous. I think the electronic health records will be much bigger, but I guess those data get, well, anonymized and cleaned a bit before we would see them. I think the imaging data, Heather can maybe talk to that later, it's ginormous, even on 1,000 individuals. Basically, we have an extremely powerful cluster environment where we run analyses, and you're quite right to flag the environmental aspect as well, particularly as we enter an AI era where we're going to throwing extremely expensive and greedy computational models at the data. It's a major, I think the cooling for the various servers and clusters is quite extensive, and it's an ongoing challenge.
Right.
We've got an awful lot less data than some people who aren't particularly interested in health but would like to troll your social media. So,
yes.
As a proportionate cost, we are a tiny, tiny tip of a very big iceberg.
Okay, so that's a positive. That positive in environmental issues. Very, very quickly, Riccardo, the aging and methylation is clearly of interest to all of us who are no longer 20. Yes, I'm sure. But my question is, we don't share. How would it look if we could share with an individual of 30 who has this environmentally clearly driven high methylation? And we know that in some percentage of people, sharing these type of data, and there is evidence, actually makes their lifestyle better, things improve, or treatments, lowering of cholesterol, blood pressure, etc., gets more adherent. Have you thought about this, and is that something to do in the future?
Thought about it an awful lot, and it's definitely something we want to pursue in the future. The field's still relatively young. And I guess these things are also what we would say probabilistic rather than deterministic. So we're not guaranteeing that you're going to get an event if you have high methylation at a certain site, but there's maybe a higher probability in the same way that if you smoke, there's a higher chance you'll get lung cancer than if you don't. But I think it's now kind of working towards ways in which how can we guarantee that the signatures we have are robust before we would then try and communicate them, and also to communicate that sensitively, because, as you say, some people will respond, other people will. Yeah.
This is absolutely great and a positive way to finish our discussion, but don't go yet. We now have, I understand, a break and photo. Is that correct? And as I said, very few of us are on this previous photo. We want everybody on the next photo, please. So you're coming back 11:40 here for the part 2, which will be chaired by Roger Halliday. Thank you very, very much. And thank you to speakers, discussants, questions were fantastic. Thank you very much. And we finished on
It won't be business today because I'm on the...
Yeah, I am as well.
All right, okay. Oh, wow. Nice. It's not going to happen. Not very far. I've got a week.
Yeah, it's hard.
No, I just wonder. It's psychological. Because everyone else is on holiday.
Yeah, I took a lot of
All right, nice.
It's hard for me to work with.
Oh, yeah, definitely so. Right. Yeah. All right, yeah. Okay, welcome back, everyone. Hope you enjoyed the break and got some good chat, and it sounds like you did. So welcome to the session where we'll focus today on the people that are at the heart of enabling all we do in Generation Scotland. The volunteers that have been part of the study and also plans for recruiting the next generation into our family. So I'm Roger Halliday. I'm the Chief Executive of Research Data Scotland, and that's an organisation that was established by Scottish Government to transform how data is used for research in the public good, making it safer, quicker, and easier to access data that improves lives. So my background is in public sector data analysis, and have worked on many studies and surveys where we rely on the generosity of the public. And, you know, I'm always impressed and that people give their time freely to all sorts of different aspects of public study. And particularly sort of conscious over the last few years that this has been even more challenging for governments, for surveys and cohort studies. And now I work in the space of administrative data, which is derived from interactions between the public and the use of services primarily. And often while there's quality problems with that data at times, and relies on general public acceptability of its use, we're fortunate in that it's got overall population coverage, and it doesn't necessarily rely on the generosity of people giving their time. Indeed, it's often by bringing together the fantastic cohort studies or surveys that we've got with the excellent administrative data that brings new insights, changes the services, policies, and leads to improvement of people's lives, which is, I guess, why we're all here today. So, you know, I'm always impressed by studies like Generation Scotland that are able to maintain the input from volunteers. So let's get started in the session by hearing about how the team are bringing in a new young people to participate in the survey, in the study. And can I introduce Heather? And Heather's a professor of neuroscience and mental health at the Centre for Clinical Brain Sciences at University of Edinburgh. And of course is the chief scientist for the Generation Scotland. And while she's got a wide range of research interests that'll take too long to run through this morning, of particular relevance here is her work understanding adolescent mental health and links between physical and mental health. So over to you, Heather.
Okay, thank you very much. That was a really great introduction. Thank you, Roger. So my task today is to talk about recruitment into Next Generation Scotland. But obviously this has been a team approach. There's been lots of people involved. I just have the privilege of talking to you about it. So many of the team are here, and it's been under Cathie's leadership, this section of the grant. So in terms of different aspects to recruitment, so the phase of the cohort, this is talking about recruiting individuals from when this Next Generation Scotland cohort began, and the recruitment really kicked off in about 2022. I'm also delighted to have the opportunity to show an illustration that was made for us by an artist called Cassandra Harris at a Generation Scotland Away Day brainstorming session. And it shows the unique aspects of the study, we think very nicely, in visual format. And the result has been a really nice figure that we are able to share. And actually the full version is down at the back on the right-hand side, if anybody wants to have a closer look at it. So the aims of Next Generation Scotland recruitment were to increase the total size of the cohort, so that in combination with the first wave of recruitment, which was between 2006 and 2011, as we've heard about, that the total sample size would increase to 40,000 participants. We also wanted to increase representation, including regional representation, and we aimed to achieve this by maximising use of remote and online methods, where people could join and complete the biosampling from the comfort of their own home. We also wanted to lower the age of that individuals could join the study. So previously that was 18 and above, and we've lowered the age to 12 and above. And this was so that we can understand the health challenges of this younger generation and follow them as they face their additional health challenges into the future. So this slide highlights one of the reasons we wanted to increase the regional representation. So you can see the map on the left. It shows the original Generation Scotland recruitment and where the volunteers have come from. And you can clearly see that there's a weighting in the central and urban areas. And this was primarily because of the necessity for participants to travel to a face-to-face clinic visit, which was the common recruitment method of the time. But with the advent of newer online recruitment methods, we saw that there were real opportunities within this next stage of Next Generation Scotland to widen participation using these remote methods. So on the right-hand side of the slide is the sign-up process that we implemented for this remote sampling. So an individual could read about the study online. They could consent to taking part if they wish to do so, answer the online 20-minute question, and then they will be sent a spit kit, as Riccardo showed in his talk, and then that could be returned by post. So all of this is online and non-invasive, and the sample is done at home and posted back to the lab. And the saliva sample kit can even be posted and travel to and from the islands without needing any special postage requirements. So this slide shows the Next Generation Scotland sample to date. So we've successfully recruited over 16,000 individuals into the study, meaning that we've reached our target of the full sample being over 40,000 participants. And due to the remote sampling, we have pretty good coverage across Scotland, as you can see from the map on the right, which looks very different to the clinic-based assessment map that I showed on the previous slide. And the graphs in the middle show how we match across urban and rural communities, where we compare pretty well to the census data. So you can see in the dotted red lines, that's the census data, and you can see that we match pretty well to that in terms of our regional representation. And the bottom graph shows SIMD levels, which is something that David touched upon. So that's socioeconomic deprivation, where 1 is the most and 5 is the least deprived. And you can see from this population that we have 25% of our new members are from 40% of the most socioeconomically deprived areas. And underrepresentation of these types of groups is often a problem with population cohort studies. So we're quite encouraged to see this, although they're still obviously underrepresented and there are still things to be done in that area. And here are some of the recruitment methods that we undertook over the course of Next Generation Scotland. And there's also a poster detailing some of these in more detail, and there's a preprint online for anybody who wishes to go and look at that. So the first phase of recruitment was what we're calling snowball recruitment. So we asked current Generation Scotland members to share information about Next Generation Scotland to their friends and family. The next is reaching out to previous survey responders. So we've heard already about the activities of Generation Scotland during the COVID pandemic. So we reached out to people who had previously taken part in these COVID surveys and giving them information about Next Generation Scotland to see if they would be interested in joining up. We also had a launch on the news media, which was in January 23, and it attracted quite a lot of interest and helped raise awareness of the study. We also engaged a social media company to put out paid social media posts, which I'll come onto in a moment. And we also created a TV advert with STV, which went out in June 2023. Other recruitment methods included the Primary Care Research Network, who were kind enough to pilot sending text messages through practices which hadn't really been done before. Share also shared information about Generation Scotland through their newsletter. And we also reached out to third sector organisations, for example, Scot Blood, Dyslexia Scotland, Men's Sheds, and other mental health charities and other disorder-specific charities, amongst many others, in order to raise awareness. And when we asked participants how they heard about us so that we could understand more about the success of different recruitment and awareness-raising routes, actually the top method was through social media. So if you look at the poster at the back, we actually had peaks for the news media and we had peaks for the STV advert, but social media was a much more prolonged kind of exposure and resulted in most of the people becoming aware and signing up on that basis. And I'm just highlighting on the right that when we looked at the different recruitment routes by socioeconomic status, we saw that the TV advertising, which is the dark blue bars, was actually much more balanced across the SES levels, so that's socioeconomic levels, than any of the other methods. Though we do note that that route was quite expensive, and it's not to say it didn't have its own biases, but it's potentially useful information for other studies wishing to kind of balance out recruitment across these levels. So I'm going to go into a little more detail about the young person recruitment. And this is quite a novel aspect of the study, recruiting from 12 and up. So this figure shows the initial young person sign-up route. So the way this worked was that a parent or guardian joined Generation Scotland. They then sent out an email if they had a young person who was interested in taking part. And then the young person would read about the study, consent to take part if they were interested, answer the questionnaire, and then complete the spit kit. But the sign-up rates for young people was quite low over the first year and a half of our recruitment. And this is something that we've heard from other studies as well, that recruiting young people into such kind of studies is difficult. So we revisited the sign-up process with our young person advisory group to think how we could improve representation of this younger generation and remove barriers. And this resulted in 4 main changes. Firstly, the young people told us that they don't use email. So these parental links were probably not being seen. Quite often in this younger age group, the email addresses for the young people is linked to a school account, so they don't check it very often. So we changed our sign-up systems to add the option to communicate with volunteers, and this is all volunteers, not just the young people, through text messaging. So when you join up, you can have the option of either communicating with us via emails or through text messages. And next, it became clear when we were going into schools, talking about the project, having to have their parent or guardian sign up first in order to pass the link on to them was a real barrier to them signing up. They wanted ownership of initiating that process themselves. So after consultation with ethics boards, we changed our systems so that the young people kind of can now initiate that process. with parental consent coming later on in the process, and there's no necessity for the parents to join themselves if they don't wish to. And thirdly, we're aware that many studies who are recruiting to young people also offer compensation for their time. And that's actually consistent with lots of funding agency commitments to research participants, is funding and compensating people for their time to take part in these kinds of studies. So we now offer a voucher for young people's time in taking part in the study, following consultation with ethics and various funding agencies. Excuse me. Finally, through going into schools, we realized that there were barriers around awareness of health research and big data, and young people being represented in these kinds of studies. And we saw an opportunity to engage with school communities to a much broader benefit for young people in terms of awareness and understanding of these topics. So we, we developed lesson plans that could be used in schools around the topics of health data research. So in collaboration with the SHINE network, and Dawn is here somewhere, I don't know if you want to raise your hands, who has been super helpful, we developed 3 lesson plans, and these were carefully matched to several parts of the curriculum and made freely available to schools. So these are off-the-shelf plans for teachers that can be used in various timetable slots like citizenship or science. And the lesson plans are around health data research, what it is, why it matters, and why it's important for young people to contribute and have their generation represented in these longitudinal population studies. So the lesson plans involve what big data is, issues around ethics and secure and safe data usage, and activities like our zombie outbreak detectives, which is very popular. And we think this is a real nice legacy for Generation Scotland, and it feels like we're actually able to give meaningfully back to the community. This resource has been requested by about 50 schools to date, which represents about 22 out of the 32 local authority areas. And we had generous follow-on funding from Research Data Scotland to continue to expand these resources. So this is my penultimate slide. So interestingly, when we asked where the young people had heard about Generation Scotland, the highest percentage was not through social media, even though through our media company we were targeting this age group and the platform that they were most likely to be on. This was only 19% compared to the 39% in the adults. So the largest percentage of responses was through family member at 41%. So we found this was quite different, quite surprising and very different to the older individuals. So in summary, we have increased the total sample size to over 40,000 participants. We've managed to increase regional representation, and we've learned valuable lessons about recruiting young people into population studies. But we're fully aware that more work needs to be done in order to engage with other underrepresented groups. And I've just put the full illustration on the right if anybody's interested in looking at it. That's down in one of the alcoves. And I'll finish there. I hope that's been an interesting overview, and thanks for listening.
Yeah, we'll move on because I'm particularly looking forward to hearing from volunteers who are participating in Generation Scotland. So, can I just introduce the chair for this bit of the session, who's Professor Blair Smith. So Blair holds the chair in population health science at the University of Dundee and works as an academic GP, worked as consultant of pain medicine and the co-director of the clinical trials unit in Tayside. And so I expect many people, though, will know him from his time as sort of co-founding and the co-investigator with Generation Scotland. So, Blair.
Thank you very much indeed, Roger. Great pleasure to be here. This session is not about science so much as participation in Generation Scotland and the reasons for it. But I'm going to start, as Roger said, I'm a general practitioner by background, and the advantage that brought me as at the beginning of Generation Scotland was, in contrast to what David was saying about all the people from different silos and different specialties competing with each other for their discipline to achieve majority. As a GP, I'm in the centre of it all, and it's all equally important to me. And I think we were able to steer successfully a course through that. And genetics at the time, population genetic studies were really on the threshold. It's difficult to imagine that only 20 years ago, and we're at this quite advanced stage of genetic research and statistical genetics. But this was just at the beginning, just at the end, just after the human genome had been characterized, and we were faced with all sorts of very exciting possibilities, which have generally come to fruition, some more than others, but Generation Scotland, I think, was really riding the crest of the wave that began there. So here we are, 26th of January 2006. This lady on the bottom left, as you're looking at it, is the first Generation Scotland participant. And this was in Ninewells Hospital in Dundee. You can see two of her family there, two research nurses, and two callow youths there who are overseeing the process. Here we are again. That's myself, and Andrew's run away, so he's not seeing this. Well, I'll make sure he sees it later. You can see Andrew and I actually first met in the beer bar in Freshers' Week at the University of Glasgow in 1982, and in first year he was the year club president, I was year club vice president, and I've always been, as you can see there, just behind him as we make any progress. So we got quite a lot of publicity at the launch in 2016, that says 2016, it's 2006, I'm sorry. I was a GP in Peterhead at the time, and at the end of a surgery, this photographer from the Daily Express came and embarrassed me greatly all across the health centre taking photographs, the one that could achieve the Daily Express's vision of what Generation Scotland could look like. But I think Riccardo spoke about the ageing process. Here we are, here various different iterations of the GS executive. By this time we've added Sandosh Padmanabhan and Lynn Hawking, who's here today. Great pleasure to see her. But equally important, the management team at that stage, Mel Foster, Archie Campbell, Robin Morton, Shona Kerr and Pamela Linkstead. And they were really the engine behind the vision that I think the people at the top had. None of it would have happened without them. Here are other iterations at various different meetings. Now, Heather, you spoke about the relaunch or the launch of NextGen Scotland, and this was, I think it was Robin Flagg who pinged me to be put in front of the cameras. This is Laura Goodwin from BBC Scotland who came to my house in January and to do a piece for BBC Scotland, which was one that was associated with the quite large jump in recruitment. Now, she began her questions by asking me why I thought Generation Scotland was important, and I explained all the scientific merits and the outputs that were going to come, and her eyes kind of glazed over a little bit. And then I said, because since we started, I've had some fairly significant medical issues myself, and I said that I might well not have been here now had it not been for people before me volunteering to participate in population studies and clinical trials. And all of a sudden her eyes pinged wide open, and I've heard since at media courses that journalists come and they're looking for the story, and in retrospect I could see that was the moment that she saw the story. And the story is that Generation Scotland and other population health studies do good work for our public health, and that was what she created, so that by the time that it came, this is a picture of my television later in the day, and I'm not categorized as a scientist or a co-founder of Generation Scotland at all. I'm categorized as a Generation Scotland volunteer. And actually now, at this stage where I am, that role is as important to me as the role that we had 20, 25 years ago in setting it up. And in this slide, you can see Generation Smith, I've called it, 3 generations of my family. There's only 3 people in that photograph who are not participants in Generation Scotland, at least as far as they've told me. And then moving on to next Generation Smith, there's now another generation below. And in fact, that's my son Blair and his daughter, who's also called Blair, actually. And in this photograph, there are 6 individuals, and they're all called Blair Smith. So that's what we're doing it for. I'm doing it for my, you get emotional now, my granddaughter and her subsequent siblings and cousins and other members of her generation. So that's what it's about. It's about learning from our parents and ourselves and passing it on down. And as we said, establishing a resource for future generations. Now, this was supposed to have been a panel discussion with a number of different participants coming up and helping to explain why they're participating in Generation Scotland. Our numbers have gone up and down. We have with us Justine Eamon, who's going to come up in a wee minute, Justine, and we'll have a discussion with him. Before that, Eva Drummond, who is a youth advocate, worked with various different charities, has a British Youth Citizen Award, was going to be here today, but unfortunately yesterday had to pull out because of health reasons. But we've been very fortunate that she sent in a short video to explain some of her reasons. I think before we put that on, we've got the woo clap that I think it was Cathie mentioned at the beginning. There's now a chance for people who have participated in Generation Scotland to help populate some of the questions that I'm maybe going to be putting to Justine later. So on this piece of paper, which was, I think, on your seat, or at least given to you as you came in, there's a QR code or a login code at the bottom. If you have a phone with you, then if you want to log in there just now and get ready to answer some questions which we'll ask. And I think online, Hannah, the QR code will be displayed online. The QR code is displayed online, so you've got the same opportunity to ask the same questions.
I'll get the video on.
Okay, so we'll get the video on. Actually, while we're getting the video on, we'll ask the first question. Is that up?
The first question's up. So
the first question is, why did you join Generation Scotland? And there are 5 chances, and we'll see how that is answered probably after the video. So thank you, Hannah.
no worries.
Hello, my name is Eva Drummond. I'm 18 years old. I've been volunteering with Generation Scotland for a couple of years now. I initially joined through a project called Loneliness in the Digital World, where we helped advise about sort of like the impact that social media and online activity can have on young people's mental health, whether there is a correlation. And from there I got introduced to Generation Scotland more widely and then became a member. But back when I joined, young people still had to have their parent to join first, so my mum is also a member of Generation Scotland. I initially joined because I was curious about what being on the other side of a health project was like. I have some chronic health conditions, so I've been a participant in research projects before, but was really curious about seeing what kind of work goes into creating one, all of the organising that goes behind it, and it was just as exciting and interesting as I had hoped it would be. I've had such an amazing experience volunteering with Generation Scotland. I've been able to do so many different things. I've learned some new skills. I've been able to go to speak at conferences across the country about sort of youth participation in health research, as well as more specifically about the Loneliness project that I was part of. I have also been interviewed by STV News when we did our little promo. I got interviewed about my participation in the project, as well as just the importance of youth participation in research. I also was able to partake in doing a participative democracy certificate with Generation Scotland, it's an accredited course that basically it meant that the work I had to put in was I had to do some presentations, I had to do some research, and I had to show that I was working as part of a group. And in the end I was able to use it on sort of like my uni application, things like that. I also was, at the end of last year in October time, I was awarded a British Citizen Youth Award, and it was Sarah, who is from Generation Scotland, who nominated me for that, and we got to go to Westminster and I got to speak about some of the work that I've been doing with Generation Scotland, and that was also very amazing. I think that health studies are really important, especially young people participating, because It helps us learn about health trends and help future generations, if not just us in the coming years, and especially for young people, that we have voices, we know what's going on with our generation in a way that older generations just don't fully understand, and also we grow up, we are going to be the future. It is going to be our health that these studies are going to impact, so I really do think it's important that we have a say and a voice in what happens during the studies and making sure that these are accessible for young people. Overall, my family is very supportive of my participation in Generation Scotland. As I said, my mum is also a member. She joined initially because she had to, because I wanted to, but also she does all of the surveys that are sent over, and we did our little saliva samples together and stuff, so she is an active member as well. In terms of my hopes for the future of health research, I hope that it is continued. I hope that there's more funding. I think that it's such an important aspect of life that people don't fully appreciate the impact of, and I hope that it gets more publicity, because especially for young people, speaking as a young person, it is such an amazing experience to be able to be part of it, as well as getting to understand how it works. It's great for uni applications, as well as just for personal development. I've been able to work on my public speaking, my teamwork, my leadership abilities. It's been such an amazing overall rounding experience that I've really enjoyed. And then finally, ways to make health research more accessible for young people. It's really simple things, if you're having meetings, make sure they're in the evenings, making sure that people can go to school, go to uni, go to college, and then can still attend these meetings. Having a little bit of flexibility, people have busy schedules, homework, exams, knowing the periods in the year that are stressful for young people, and understanding that participation may drop around these periods, but that doesn't mean that young people aren't interested. It just means that unfortunately we do have to prioritise other things. Other things like using appropriate language, making sure that there isn't so much health speak, making sure that cause for older young people, I would say probably from 16 to 25, for the most part, health language, scientific language is fine. But if you are looking to interact with people younger than that, they might not always understand what that means, and then also might not always feel comfortable admitting that they don't understand what something means, which then creates a barrier that you won't be aware of, and that young people would be too afraid to speak about. So just constantly making sure that language is appropriate, things are explained, even if nobody asks you to explain them. And then finally, my biggest tip would be to treat young people as if the same that you would any adult participant or health research, researchers, anyone at uni, that kind of stuff. We have the same capabilities as those people. We may not have the same expertise, but we are able to articulate our thoughts, we are able to express our opinions, and it's really important that people feel like they're listened to. That's what brings young people back, that's what encourages them to keep going. If you feel like you're constantly being shut down, then you're never going to keep going to something or keep participating, and it is really important that young people keep doing this and keep volunteering and keep participating. So overall, I've had the most amazing time working with Generation Scotland, and I can't wait to see what the future for Generation Scotland holds. Thank you, bye.
So there's the outcome of the first question from the participants. So you can see that it's changing. Ooh, dynamic. It looks like you could only tick one box, and I guess that people would maybe tick all four, but the most important reason participants here and online are participating in or joining Generation Scotland is to contribute to scientific research. We'll maybe move on to the second question. And just before we introduce Justine, second question is, why is health research important to you? And I'll give you a minute to think about that. And while we do that, it's my great pleasure to introduce our panellist, who's Justine Eamon, a strategic project manager he wears a number of different hats, but he's here representing, I think, Passion for Fusion, which is an organisation promoting the interests of black and multi-ethnic communities, striving very much in line with what Heather was presenting here, striving for an inclusive and equal Scotland where everyone is empowered to participate in cultural and multicultural Scotland. Justine is here as a participant in Generation Scotland. That's the main hat he's wearing today. So perhaps we can invite you to come up and we'll ask some of these questions and get you to explain yourself. So if you'd like to take a seat, Justine. You can see the results of the dynamic questioning behind you, but we'll start off, Justine, maybe you'd like to introduce yourself, and in the context of the first question I'm going to ask you, which is the first one we asked everyone else, which is, why did you join Generation Scotland?
Okay, great. Thank you very much. I'm audible enough? Okay, great. Firstly, I would say it's a big honor to be here today, and seeing the number of people in the room just now tells me the impact, the great work Generation Scotland has been doing is really appreciated and supported by people from various walks of life. So I would say big well done to Generation Scotland and big well done to everyone in the room just now as well. And about myself, I think a bit of it has been said already.
Okay.
Yeah, so I work for a tech sector organization called Passion for Fusion. And the organization is based in Edinburgh and in West Lothian as well. We are involved in so many projects that support people from diverse ethnicities, especially those from deprived communities, irrespective of their original backgrounds. Yeah, and our work generally is divided into two parts. We've got young people and children work, where we do robotics, STEM, creative arts, dance lessons, and so many other, including sports as well. And we also do adult and parent work, where we support people into work, employability, the Promise Scotland, and so many other stuff that generally supports the entire family. And that's one of the reasons the work with Generation Scotland resonates so well with us. Yeah, because at the end of the day, it's all about making everyone to be healthier, happier, and live longer. Thank you very much.
So, thank you very much for that. What was the process through which you came to join Generation Scotland? How come you were recruited?
Yes, the process for me was really easier. And that's one of the reasons I not only joined, but I've chosen to be with Generation Scotland for the long term. Yeah, because my family too liked the process. It was so simple and seamless. I got introduced by a friend, very good friend of mine, which most of us, if not all of us, already know, Sarah Robson. Yeah, so she visited our youth group.
I see.
Yeah. During one of our activities, and she talked to us about it, and we're like, yes, this is a great opportunity to contribute to health research that is meaningful, that is relatable.
So Sarah came to speak to you, but possibly with her main target being the youth group that you were running.
Yeah.
And what I'm seeing is a parallel, because initially I participated in Generation Scotland and twisted the arms of my family too.
Okay.
Because as a researcher, as a clinical researcher, I've always thought it's not fair for me to ask other people to do things that I'm not willing to do myself. Partly because just that's self-explanatory, but also partly so that I can understand the experience that people are getting. And I wonder if that was the same as you. As I said, Sarah's target was the youth group, and not saying that you're not a youth yourself, Justine, but was there any sense of that with you?
Yeah, well, on a lighter note, physically, I'm not a youth, but in my heart I am.
Yeah, good for you.
Yeah, because I work with young people every now and then, and it's been an exciting journey for me. So anything that interests the youth, I kind of resonates with me as well, because at the end of the day, we need to give them every support that they need to help them go into the future well prepared.
Okay.
Yeah.
And in that regard, I wonder how you think Generation Scotland specifically can support the youth in our country as they do move forward.
Okay.
What do you...
Yes, for me, I think Generation Scotland is doing amazingly well already, but of course, the biggest room in the world is the room for improvement.
Okay.
So there's a need to continue to improve, especially in the way young people are recruited. Yeah, making it even more relatable to them. Yeah, not just within the four walls of a school setting alone, but through other avenues. I like from the survey that was shown how recruitment was done through social media. Yeah, among other platforms. Yeah, social media these days seems to be one of the best ways to get the interest of young people. Yeah, so, but the content in social media that appeals to an adult may not necessarily appeal exactly the same way to young people too. So our content to gradually be fine-tuned even more to attract the attention of more young people.
Right, so, and I wonder, Hannah, if we can change and put the third, we can see the results of the first one, the second question. Sorry, before we move, can we get the third one up in a wee minute? But, okay. The main reason for why health research was important, you could see, was to improve health outcomes. But if we put the third question up, which is what inspired you to become involved in health research through Generation Scotland, which is kind of the question I've asked you, Justine. I think to me, your answer, one of the main reasons there was about kind of what you might call citizenship. It's about being part of society and democratizing scientific research and participation generally. And I wonder just how that resonates with other participants, because what you didn't mention, and I'm sure you would have done had you come around to it, but was the specific scientific outputs. What are we discovering about health and illness and potential treatment through your participation? Was that something that motivated you alongside the citizenship?
Exactly, it does. It does as well, because, excuse me, it's so important that the root cause of illnesses are discovered or researched upon and backed by real evidences. Yeah, so for me, that's one of the reasons why I feel this is a brilliant idea to be part of. You know, not just coming up with research results only, but having real evidences. Yeah. And the entire work of Generation Scotland embodies this. There's always evidence for every research result.
And I suppose specifically with the young people that you're working with, taking the point that David Porteous made at the beginning, which nature versus nurture. Yeah. If we remove the versus, it's nature and nurture. And what we can learn from a genetic environmental study like this, particularly of relevance to young people, is how the nurture, how their lifestyle may contribute to their long-term illnesses, whether that's cardiovascular disease, cancer, or even just the aging process. So that's the opportunity here. We'll move in a wee while, Hannah, to question four. But I'll get out of the way because I'm blocking what inspired you to become involved in health research through Generation Scotland? Well, with me, it was the first of these, curiosity about genetics, when I was reading about the human genome. Interestingly, it's influence from an academic or professional background of the participants, at least here or online today. The other important reason is the desire to support community health. I mean, I think they're all good reasons, and it would be interesting in due course to read about the other ones, but we don't have the opportunity here. If we go to one which might need a little bit more thought just towards as we reach the end of this session, the question is what areas of health research, what areas of research, that's the one probably. We've got a few questions, we don't have time for them all. But what areas of health research would you like Generation Scotland and researchers to focus on in the future? And this is asking for free text words rather than a multi-choice response. And we'll show that in a little while, but I wonder, Justine, if you, from your perspective, or from the perspective of the constituency which you may represent, Passion for Fusion or youths or general population, what areas of health research do you think it's important for us in Generation Scotland to research?
Okay. For me, of course, so many areas to recommend. Yeah, so but one or two actually stick or pops at my face almost every now and then, and that's in the area of both physical and mental health. Yes. Yeah. With particular focus around preventing and early intervention.
Yes.
Yeah, so I think a combination of these two in the would go a long way to minimize a lot of the issues that are beginning to come up that people go through. Yeah. And at the end of the day, prevention is actually cheaper.
Yes.
Yeah, preventive health is actually cheaper and easier, so to speak. Of course, nothing good is always as easy as, but comparatively, I think it's better to look at preventing and early intervention, both physical and mental health.
Yeah, thank you. And I think that's a philosophy that most of the scientific team in Generation Scotland would concur with, particularly Riccardo, for example, was showing us the benefits of early detection of subsequent potential long-term harms. So if you represent the general public opinion, which you're not being asked to, then that's something I think we can work on. And you can see in the word cloud that is developing behind us. I'll come down here, hopefully not trip up. The big words, mental health is there, dementia, which crosses over. The word chronic, and I saw prevention in there somewhere. Women's health, of course, is has always been important, but relatively under-researched, so it's important to correct that balance. It's interesting. I mean, I'm not going to read it out to you. Hopefully you can see it yourself. But I think chronic conditions, mental health, dementia, women's health seem to be balanced in these. I would like to, my main research area is in chronic pain, so I'd like to think that is included in that word cloud. And I would agree with you, Justine, that prevention of the impact of chronic pain is far preferable and potentially easier than waiting until it has established itself. So all very interesting, and thanks very much to Hannah for your technical expertise, but a particular thanks we'll draw to a conclusion for this part of it. Thank you for your participation and for listening, but particularly thank you, Justine, for joining us here and giving us the benefit of your experience.
Thank you so much.
Great. Wow, what a session. A couple of themes from me are, it's first of all, representation, really important for studies to make sure that they're applicable to a wide range. So it's really impressive the work that's been done to get more representation across the country, across so much demographic groups, particularly around young people. And impressive contributions from Justine and from Eve there on sort of feedback on, actually, how do we recruit, how do we nurture the participants? And also, thank you, everybody, for the contributions that you made to the polls. Some real clear themes there about the motivations that you've got, and actually the wide range of research themes that you're looking to try and influence. And looking through the Generation Scotland website, can see that points to the massively wide range of themes that are there. In fact, after lunch, when we come back at 1:35, we're going to be focusing on the research that's done. So that's a really nice setup there. But I'm left with the line that Blair gave earlier on that, might not be here without the volunteers that have dedicated time and given their time generously to health research. And so, that's really where it is from me. So thank you for the contributions that you've made, and that's why we're in this, right? So enjoy your lunch, and we'll catch up in the afternoon. All right, thank you.
Okay, so I think we'll get going again. So we heard a little bit in this morning about some of the biomarker work that's been going on in Generation Scotland, and we're now going to hear about some of that in more... Oh, you can't hear me. That better? Are we on? Just to... Okay, yes, so thank you. So we heard a bit about biomarkers this morning. We're now going to go... Sorry? We're going to hear some more now. So first to introduce a senior research fellow from the Division of Psychiatry, so Xueyi Shen. So she has been working a lot on inflammation and depression. So, over to you, Shen.
Thank you, Riccardo. It's much louder for me because I'm shorter. Good afternoon, everyone. My name is Xueyi Shen. I'm a senior research fellow here in Edinburgh, and it's my great pleasure here today to talk about our work on DNA methylation, depression, and genetic risk for depression. So World Health Organization predicted that depression is going to be the single biggest lead to disability worldwide by the age of 2030. In a report by NHS last year, over 8.6 million people are currently taking antidepressants, and more than 1 million of those individuals have been taking antidepressants for a very long time, for over 5 years. Although people are taking antidepressants, but they don't really respond very well. According to a study in 2023, around about 30% of antidepressant users actually do not respond to treatments, and this prevalence can go up to nearly 50% in some areas of the world. These are some grim facts. They suggest that depression is very common, is very disabling, and is really difficult to treat. And all of these facts point for depression to be a high priority for health research. In order to help us understand and treat depression better, novel molecular findings are very much needed. Some advances have been made on genetic studies for depression. For example, based on twin studies, depression has a heritability of 27%. This is relatively moderate compared to other heritable traits, such as height, which has a heritability of over 80%. So what does this heritability mean? For example, when we look at identical twins, although they have nearly identical DNA and they have very similar upbringing, however, some individuals can grow to develop depression later in life, whereas their twin siblings can remain completely healthy. And this difference cannot be explained by DNA alone. So how do we understand this puzzling difference? One way to understand this is epigenetics. We know that the genome isn't just DNA codes, as introduced by Riccardo in the morning. Some epigenetic markers are attached to DNA strands that can help alter gene functions. One of the most well-studied epigenetic marker is called DNA methylation. Those methyl groups are attached to DNA strands without changing its sequence, and they can alter gene expression. It can turn genes on or off, or alter the magnitude of gene expression. So overall, DNA methylation is an interesting marker. It can be influenced by many different things. We know that DNA methylation can be influenced by genetic itself. It is also very sensitive to the environment, for example, diets, smoking, alcohol consumption, or trauma. So DNA methylation is an attractive marker, especially for depression, to help us understand depression, especially the bits that is not completely explained by genetics alone. Generation Scotland is an amazing resource. It is actually the biggest DNA methylation data in the world so far. This really gives us a strong position to do analysis to test our hypothesis linking depression and methylation. So here we used the first release in Generation Scotland, including 2 data sets. The aim of this study is to use DNA methylation and machine learning algorithms to generate a score and test whether this score can help differentiate depression cases from controls. So here in this study, we used around about 3,000 people to train the data using machine learning algorithm. and then we generated methylation scores in the testing data set. To help us understand whether the methylation scores perform well, we also created polygenic scores, which is a well-established benchmark score. This score was trained on a much larger GWAS study from 800,000 people. So here is what we found. We found that both methylation scores and polygenic scores were able to differentiate MDD cases from controls. And surprisingly, they perform quite similarly, despite the fact that methylation score was trained on a much, much smaller training data set. And we then combined the two scores in the same model. What we found was that the combined performance of two scores is better than one score alone, meaning that they have distinct features to help us predict onset of depression. So overall, in this study, we used machine learning to create methylation scores, and they were able to help us differentiate MDD cases from controls. And there is some shared and distinct mechanism between genetics and DNA methylation. However, in this study, we took the entire methylation as an overall signal. The next natural question is, can we identify a specific genomic locus that is associated with depression? Some researchers tried to answer that question using smaller data sets. The largest study prior to us looked at almost 8,000 individuals, and they only managed to identify one CPG site associated with depressive symptoms, and they were not able to validate it. So what we learned from this study is that we need much larger, big, reliable data sets to help us reveal meaningful associations. What we did was that we combined DNA methylation data from 18 different cohorts, including Generation Scotland, comprising nearly 25,000 individuals. We identified 15 CPG sites associated with depression, and they were validated in an independent cohort. Looking at what these CPGs do, 3 of them are either associated with MDD diagnosis in previous literature or associated with response to antidepressants. What's really interesting is that some other CPG sites map to genes that are particularly important for immune functions. They're associated with markers such as LDL cholesterol levels and C-reactive protein. So here we found some associations, but we wanted to know if there is any causal effects. We used a genetic tool called Mendelian randomization. It leverages genetic variants to help us identify a potential causal mechanism. What we're really interested in here is whether there's any causal effects coming from DNA methylation to major depression. We used 2 separate data sets. First, we conducted a discovery analysis using Generation Scotland, and then we replicated the findings using data from a cohort called GoDMC. Here, we identified 23 potentially causal effects from DNA methylation to depression, and the vast majority of signals were in the MHC region. We then replicated the findings and found that 6 were replicated in an independent analysis, and they were all located in the MHC region. So you may ask, after a very long lunch with carbs, what is this MHC region? MHC region is one of the first genetic loci identified and replicated for psychiatric conditions such as schizophrenia and MDD. It is a very big and complex region with lots of protein encoding genes. So proteins encoded by genes located within this region are very well known to be associated with immune functions and inflammation. This is not the first time we identified association between MHC and depression. In another study, instead of looking at MDD diagnosis, we also looked at genetic risk for depression. As you can see in this plot, we found a very large and strong signal, again in the MHC region. We then sought to replicate these findings in 2 replication cohorts. On the left-hand side shows you the replication in an adult cohort coming from low-thin birth cohorts, And on the right-hand side shows you the replication in an adolescent cohort with a mean age of 17 from Alsbach. What we see here is that the signals we discovered in Generation Scotland were very well replicated in both Lothian birth cohorts and Alsbach. This means that not only that we validated our findings, but also these methylomic signatures may occur very early in life. So in summary, through these series of studies, we found replicable DNA methylation signatures for major depression and its genetic risk. Those CPG sites we found were closely linked with immune response. So these findings really support an increasingly popular theory called the immune depression hypothesis. This was beautifully described in a book called The Inflamed Mind by Professor Ed Bulmore. And by the way, this book is, if you get 3, you can get another one for free, so it's a good opportunity if you have a group of 4. This is based on observations from Ed Bulmore, based on his own experience as a clinician in Cambridge as a very young psychiatrist. What he found is that depression very often co-occur with immune dysfunctions. For example, if you have arthritis or if you're experiencing recovery from a major surgery. He was very excited about the finding, but for a very long time, people have been believing that immune functions, immune disorders, and depression, they're very separate things because of a mechanism in the brain called blood-brain barrier. This barrier protects the brain from inflammations happening in the body. But latest findings suggest that this blood-brain barrier is not that strong and tough. It could be more vulnerable than we thought. And here in our finding, we also support this theory, which provides exciting opportunities for example, identifying subtypes of depression or drug repurposing opportunities. So here I would like to thank all my collaborators in Edinburgh and from PGC MDD Working Group. Special thanks to Professor Andrew McIntosh in Edinburgh and Professor Naomi Ray from Oxford for their guidance. Special thanks to Generation Scotland, which provides a very strong position for us to lead these very large international collaborations. Thank you.
Thank you so much, Shen. So we're now going to move on to a talk from Naveed Sattar. So Naveed is Professor of Cardiometabolic Medicine at the University of Glasgow. He's been a long-time contributor, user, collaborator of Generation Scotland, and he's going to tell us today a bit about some of his work on proteomics. So, Naveed, the floor is yours.
Okay, thank you very much. I see the rain is here in Edinburgh, and it was beautiful when I left Glasgow, but it's always like that, I'm afraid. No, I'm joking, of course. So really, I wasn't really proteins that are in the blood, so that signal health. And I think, obviously, I wasn't here this morning. I was doing my clinic this morning, but I guess in measuring various proteins to look at risk, and I'll discuss that briefly. And I think, Riccardo, you've done some of this in the morning as a kind of pressy. So here are my disclosures. So I probably one of the most important ones. I do this thing called, the pointer doesn't work, never mind. Oh, does it? No, it doesn't matter. It's too, does that work? No, doesn't matter. So I work for the UK government. Most of my work nowadays in research is on obesity, of course, which is a major problem, and the major driver of this is effectively far too many calories in society that are relatively cheap. So I work with lots of different companies. I have no shares in any of them, just to for open disclosure. So, in the clinic this morning, the conditions that I treat, we all worry about premature death. None of us wants to die too soon, I worry, the other conditions that if you put surveys up, is usually cancer, dementia, and stroke are the things that will really worry people because of the things that, you know for obvious reasons. But of course, other people, we also worry about heart attacks. No one wants to have a heart attack. Any of these conditions have major impacts on our lives and maybe our functional capacity as well, so they really do matter. For heart attacks and strokes, as many of you know, if you come to our clinic, if you go to any clinic, your GP, he has in front of him, we don't use these kind of charts anymore, but we have a risk score. So if you tell me your age, I can see whether you're male or female, and whether you smoke, your blood pressure, your cholesterol, your HDL, whether you've got diabetes or not, and plus a few other things, we can, with reasonable accuracy, give a sense of what your risk for heart attacks or strokes are over the next 10 years. Reasonable accuracy. But it's not perfect. It's by no means perfect. And for years we've thought, can we actually make this better? Because when we know your risk, one of the decisions we have to make is, should I be treating you more, should I be recommending, should we have a discussion with you as a patient about whether to start a statin treatment? And many have read about statins in the Daily Mail, and you think, oh, they're really bad. Well, actually, they're not really bad. They're pretty damn safe. They're probably about 500 times safer than aspirin. But it's still a big decision to go on a medication, because there's implications for you if you've never been on a tablet for being medicalized, has implications for your insurance and multiple other things. Equally, blood pressure should also be dictated by not just your blood pressure level, but your risk for heart disease. So, and we've now... Oh, does this start working for some reason? I don't know why it's not. I'll just do it this way. No, it's dead. It's died. Why is it died, Riccardo? Ah, there you go. So, we have a couple of proteins that we thought we would measure in Generation Scotland. One of them is called troponins, there's two different types, and there's another one called NT-proBNP. And of all the biomarkers that Riccardo and other people in the world can measure proteomics, thousands of proteins in your blood, the top two that come up for heart disease and stroke nearly every time, or heart failure, are these two. Nearly every proteomic is these two markers. Nearly every single marker. Troponin is actually released when your heart gets damaged and the cells break up, and troponin then comes out into the blood. And if you've got chest pain and you're breathless and you've got pain and we measure it's really high, you are having a heart attack because your heart is releasing this stuff in really excess and you're having a heart attack. But we've now discovered that even when you're not having a heart attack, you can have low levels of this excretion, which tells us about your future risk, because you're maybe getting a bit of ischemia that you don't know, i.e., some part of your heart cells are a little bit stressed, even when we're all sitting here. Some of us might have slightly high troponin levels. Mine's slightly higher than Cathie's, I can tell you that for sure. Right? I'll tell you why for later. And the other one is actually... The heart is working, it's not because I'm up here, it's because I'm Glaswegian, of course. The deep-fried Mars bar. Anyway, the heart is working too hard or under stress. Now, this is not the heart actually breaking down in damage. It's a kind of volumatic, or it's just working too hard. There's no particular damage to within the cells, but something to do with stretch of the cells of the heart is making you release this. And that protein, NT-proBNP or BNP, is released in part to protect you, but as it goes really high, it tells us about your risk. So we're capturing risk from these two things. Paul Welsh deserves, he's not here, he's my brilliant colleague. We work with Riccardo and myself and Caroline, and Archie's here. Kind of led a lot of this work with me, and Paul deserves great credit. He came as a postdoc having done immunology, but then he saw the light. I'm just joking, actually. And I'm just going to briefly, heart attacks, troponins, broadly, maybe the major, some of the major risk factors, particularly when you went to the 1990s, were your LDL, your bad cholesterol, and smoking. Whereas heart failure is not so much linked to LDL, it is linked to a bit about smoking, but it's also more driven by alcohol. I saw a lady this morning who drank, she said, I don't drink that much, Doc. I only drink a bottle of wine at the weekend and maybe another couple in the middle of the week. I thought, oh, right. And her BNP was actually quite high, and she had obesity. And I thought, you can integrate lifestyle with risk, and it all kind of starts to make sense. Other factors that often you get with heart failure is poor kidney function. And there's a stress that you have with volume and other aspects that affects your filters, which is your kidney, and your pumps and your pipes, which is your heart. That's what happens. Anyway, we measured troponin in the two types in Generation Scotland because people didn't really know which is the better in low grade. So most people, this is measured when people were not, when they were stable. So nobody was having a heart attack. The levels for heart attack are much higher. But we discovered there's a range between sort of 1 and 20. even in the steady state, even within us now, if you measured it, there might well be somebody in here whose level might be 20, probably me, okay? I have a family history of diabetes, I'm Glaswegian, my brother's had heart attack in his 40s, my dad, you know, that kind of, so I've got risk factors. I might be very handsome, but something's, anyway, I'm deluded. But seriously, if your level is an average of 10, then you have a 10-fold risk of a heart attack or stroke over the next 10 years, compared to someone whose level is under 1. Wow, 10-fold, that's bloody high. Okay, we haven't adjusted for other risk factors. When we adjust for other risk factors, so just for your age and all the things that might create that environment, your risk is still about double. And we discovered that actually you might be able to improve prediction of risk by about 7%, which is kind of modest. Anyway, and other people have said, well, actually, can we then use troponin in those people in whom, in the middle, I'm not going to go through this, with the risk score, if your risk is really low, it's always going to be low. There's no point in doing anything else. If your risk is really high, well, you're high. You know, I don't need to do any fancy imaging or other things. We know it's high. Statin, blood pressure, smoking. It's the bits in the middle where we're not really sure. Is there something else we should do? Should we do an image, which Edinburgh colleagues are trialing? Should we do another blood test like troponin? And can that help us give us better information? And that's the kind of paradigm. Well, now, we're not quite there yet. The jury is still out on troponin, and actually we published a recent paper with lots of cohorts, and I still think the jury's out. And the issue is troponin is not cheap. It's not expensive, it's about £3, but when you think a lipid level is about 1 to 2 pence, and £3 times a lot of people is a lot of money, okay? So there you go. Now, BMP is the other one, stretch, and we were able to develop a reference range, which Paul led, and the solid lines tells us about levels in men and women. The women's actually slightly higher than men. And actually, before the age of 40, women's levels tend to be higher, which is kind of bizarre because men get more heart disease and men get more heart failure. Something funny going on. We don't fully understand this protein in every sense, but we do know, and the broken lines are the top 2.5% in the population. So normally, if you're sitting here healthy, don't go out and rush and get this measured, okay? But if I see a patient, you know, this lady this morning's level was 279, which is kind of pretty high. I would love it to have been 5 to 10, but with age it goes up. But if you see a level of 500 or 400, then I will go and get an echo. If you see a level of 1,000, then that lady or individual has probably got heart failure. If you see levels of several thousand, they're probably symptomatic heart failure. See how fast it can go up exponentially. And we then put a reference range, and I'm not going to go through this in any detail, but it's been well cited. And the beauty of Generation Scotland is that lots of samples across a range of people that allows us to look at age, sex, and different aspects. And I think this paper's been cited about 40 times already, even though it was published a couple of years ago, and people are looking at it and think, oh, wow, we did a lot more in depth. Right, how am I doing for time, actually? 5 minutes? Okay, I've only got 3 or 4. Well. GDF15 is another protein which is coming in. We also measured at the same time because Roche gave us the assays, and I said, would you give us that assay at the same time? We'll measure these things. They said, okay. And I wasn't too excited, but it's a marker that, if you haven't heard, it's related to stress, inflammation. The disease comes out of cells. So kidney problems, cancers, inflammation, back to inflammation, and lots of, any type of stress in cells can push it up. Early detection of conditions can we use it for? Can they help to progress the conditions? And it actually goes up really high in cancers, and it's probably the protein that causes people to get cachexia, stops eating, we think. Again, we were able to develop a reference range. We put it in a modest journal with an impact factor of, we get crazy about 3 or 4, but it's been cited 36 times because it's become really exciting as a target for preventing people losing weight in cancer. Wow. And there's a reference range, but we then also looked at it in relation to heart disease, and to our surprise, now look at troponin. This is the higher the level, the higher the risk of heart attacks or strokes over the next 10 years. And troponin levels we thought were good. We thought they should be the next best thing. But look, NT-proBNP and GDF15 predict the risk of heart attacks and strokes better than something released directly from the heart. Wow, what the hell's going on there? So we don't quite understand that, but that's kind of interesting. What we would like to do, and actually I had a thought about this, we also know that when you get heart disease, there's a higher risk of dementia and cognitive decline. We've already published that troponin and BNP predict cognitive decline. And we're going to do some work with colleagues in Edinburgh, so whoever's leading the dementia mission, and I think, Andrew, you know about this, and we're going to do this. We should probably look at these in Generation Scotland as well, and in due course. And then in a side issue, GDF15 is such a really amazing, complex molecule. So we've got the best reference range in the world, and people are now citing that. And Caroline's here. I was working with Steve O'Reilly. We discovered that metformin, drug in diabetes, causes kind of tummy ache and slight upset. And guess what? It increases GDF15. And we think that's probably the mechanism. We published that in Nature. And at the same time, Steve O'Reilly said, oh, Naveed, there's something about hyperemesis, women who get sickness in pregnancy. It goes up, and GDF15 goes up. So using stuff that Steve did, very clever, other stuff we had, and then Caroline in Generation Scotland, I thought about it. So I thought, wait a second. We know women who've had hyperemesis I think, in Generation Scotland. We can look at genes and look at their levels. And we did discover that women who tend to have low levels of GDF15, when they encounter pregnancy, the fetal placental unit makes GDF15. So if a woman is used to low levels and the levels go up much, much higher because of pregnancy, they get really, really sick. So this paper is suggesting that GDF15 might actually be the cause of hyperemesis in pregnancy. Wow. Bloody wow. And not only that, and I wrote to Steve, I said, Steve, I wonder whether we could give metformin to women pre-pregnancy who've had hyperemesis. And Naveed, I've already patented that idea. I thought, Steve O'Reilly's really, Sir Steve O'Reilly, you know. And that's been cited 114 times, and Caroline had a great role in this. And, you know, it just shows you the value of doing something really well in big numbers. And I'll just skip this for time, but, you know, if you want to look at proteomics, we can measure lots more proteins. And different patterns of different proteins tell us about risk of different things. But the more proteins we measure, we can definitely improve prediction of different things. And we tried to get proteomics in Generation Scotland. Have you got O-Link done yet, Riccardo? We tried to get a grant for that 15 years ago. And they said no. Anyway, it doesn't matter. So, I know, but never mind. And instead, they went and gave it to UK Biobank. I blame Cathie. No, I don't blame Cathie. Just So I'll stop there. I want to thank all the Generation Scotland participants. This is just a small snippet. It's allowed important research and some of the most compelling blood tests in areas of need that predict heart attacks, strokes, heart failure, cancer. The impact has actually genuinely been worldwide and ultimately, hopefully, should lead to better future health for many people. And the most important thing, it's allowed Glasgow and Edinburgh people to speak together. It's amazing. I thought you guys all had horns, but actually you're quite nice. So anyway, I'll stop there. Thank you very much.
Thank you so much. We should have some mics for Naveed and... Yeah. Questions from the audience, questions online. I'll maybe start with one. What is it, Naveed? GDF15 is fast becoming my favorite protein. It's so interesting. Yeah, we looked at it in the UK Biobank, and it was associated with incident dementia, COPD, all sorts of diverse diseases. Question though, Naveed. Do you think it is more likely cause or consequence?
Sorry, say again.
GDF15, cause or consequence?
Of what?
Disease. It associates with so many different diseases.
So
would treating it make a...
Yeah, no, I think it's a bit of both, actually. It depends what you're looking at. It's always about outcomes. So there is a possibility that GDF15 is a causal element in cachexia, hyperemesis, and in cancer. And in fact, they've done an anti-GDF15 trial in cancer, published in the New England Journal, with a drug called Prograbimab or something, which actually showed a reduction in cachexia. Now, one of your former colleagues who's now gone to Oslo, Barry Laird, told me, maybe I shan't say this, he reviewed the paper. He thought it was reasonably promising, but we need a few more trials because one swallow does not make a summer. But it looks like blocking GDF15 in the context of cancer can suppress or can mitigate cachexia. Now, for other things, I think it's probably a marker because it's reflecting stress in various tissue factors. Like heart failure. Is GDF15 a cause of heart failure? Nah, don't believe it. Of course it's not. But it's high in heart failure, but again, when people get severe heart failure, they get lots of inflammation. Some of the stress might be that the cells, when they're really distressed, and they're pumping out not only IL-6, but they're also pumping out GDF-15, which is causing cachexia, because you get cachexia in the context of heart failure as well. So,
good stuff. Any questions from the floor or anything online? Oh, David. Oh, Nic first.
Nic Timpson's not allowed to ask. No, yes, that's That's okay. Good. Yeah. Can I predict what you're going to ask? No, go on.
Can I just ask a question going back to the issue of depression, and ask, now that you've been able to see this very strong association with MHC locus, if you compare individuals with depression with or without MHC as a risk, genetically observed risk factor, do you see any promising or obvious differentiations between the type of depression, age of onset, drug responsiveness, etc.?
Thank you for the question. Just to say, I didn't know there's a Q&A, so I may be just talking really rubbish here. But from what we recall, I think there is some initial work on a subtype of depression. They call it atypical depression, and people tend to eat more, sleep more, rather than eat less and sleep less, like the typical ones. And they often have these immune problems, and they tend to respond much worse than people, the normal typical ones. So I think there are some initial works, and I think there are some randomized controlled trials out there testing whether, for example, cardiovascular drugs can actually help to cure depression along with antidepressants. But it is definitely ongoing work.
Can I nip in before Nic? So, depression prevalence, is it going up substantially at the moment? And if so, sure, that cannot be genetics. So presumably it's environment, maybe iPhones, all the anxiety that's happening in the world, changing lifestyles for young people. Is that a big factor these days?
I think it's Trump for depression instead of iPhone. No, I'm joking, sorry.
You said Trump.
Are we online? So, from what I know, yes, I think there is an increasing trend of depression, but this is very difficult to tell why there is, because I think compared to cardiovascular conditions, depression is mood, and it's
subjective.
Yes,
part.
yes,
Okay.
exactly. Sometimes it's because of raised awareness, people realize that
Sure.
they're having depression, and some exciting work are going on in Africa, led by Andrew McIntosh. So I think in some areas where there's lower prevalence, we're really making effort to study those populations
Okay.
and raise awareness.
Okay. Can I sort just before Nic, I want to shout out to David as well. Sorry, David Porteous, who was really important to some of starting our interactions. Sorry.
Okay, Nic? Oh, microphone's on its way.
Thanks. Two terrific talks. The MHC is fascinating because it's also hyperstructured, so it varies. I guess for the audience, it varies in a way which is correlated with demography and how populations vary. And I just wonder whether you could comment on how that might influence the way you interpret this fascinating connection between the epigenome, which we know responds to the environment, all those other things that can have an effect on depression. Naveed's just mentioned some, you've mentioned a certain person in the States. And then there's this other element, which is genetics. But actually the genetics you've got might be part of the same story. I just wonder whether that's part of your thought.
I'm not surprised that you asked this question as a PI of ours back. I do agree. There are loads of environmental factors that could contribute to either gene functions within the MHC region. I think loads of studies, for example, your early life trauma, and smoking environment, not just yourself smoking, but within the environment, those environmental factors could influence gene expression of MHC. But I guess I'm also interested in more broader environmental factors such as poverty and educational attainment, whether those could influence immunology and their link with depression. I think that's interesting. That could be a next paper.
Can I just make a comment? So I've just written a comment in the Lancet Diabetes. We have this same thing about obesity, and Nick and I have this discussion, and the genes, I asked this, actually I got this from Nick, so if it's wrong, he's to blame. So the genes for obesity haven't changed over the last 50 years, but that doesn't mean to say they're not important. It's that the environment has changed so that people who are susceptible perhaps now are, with more food in the environment, are more susceptible to putting on weight. And it may be similar in depression, is that you've got these genes plus gene-environment interactions and epigenome, so that suddenly with changes in the environment or whatever, news, social media, phones, whatever, lack of sleep, is putting some genetic susceptibilities to the fore, and that's leading to it as well, plus greater awareness. But certainly for obesity, it's not about willpower predominantly, it's predominantly about the environmental changes, I think, for obesity. You can ask me really
Yeah. This is not a revenge. No, I'm joking. So I'm really interested in proteomics, and I think there is some quite exciting work differentiating protein levels in different cells and single cell studies. Do you see opportunities of...
I have no idea. So Riccardo, pass it to the chairman. That's what you should do. Pass it to the chairman. Single cell...
Proteomics, I guess it's very much in its infancy. I mean, it would be fascinating to look at. I still think there's an awful lot we can do with bulk, to be honest. I was going to answer a question by asking a question. Have you done anything with either depression or the cardiometabolic diseases where you've, within people who go on to develop the diseases, find proteomic signatures or data-driven clusters? So do you get subtypes even within those who go on?
I can answer. So there are. I mean, Nick's probably been involved in. So there are subtypes. So our colleague Ewan Pearson has done some of this actually in diabetes with subtypes of type 2 diabetes, and there are people who. So if you think about type 2 diabetes, it's a disease predominantly of excessive topic fat, but some people put it on, develop diabetes at lower BMIs, particularly, and you see that in certain groups, white individuals, usually because they've got low muscle mass or low pancreatic insulin production, and they need less obesity to trigger it. Other people need a lot more obesity to trigger it. Some people develop it young, some people develop it old. So there are variances in some of the factors that lead to diabetes, that leads to heterogeneity in diabetes presentation, which we can find, and it's been picked up by clusters. There was a classic paper by it was the Swedes in Lund on clusters. Some of it wasn't right though. If they don't, you've got to match for various factors. Some of the clustering is just by variance of age presentation and other things, so you've got to be very careful. And so I think some of the clustering is definitely worthwhile, some of it's overhyped, and I still think we're searching for the truth.
So as a follow-up to that for both of you, how good do you think are ICD codes and read codes, so hospital and GP codes, in terms of actually classifying people with a specific disease to begin with?
Do you want to? I mean, I can tell you, for some conditions, it's really good, because if you've had a heart attack, it's a hard
obvious,
event.
But it's all...
Yeah. And for diabetes, it's not bad, because most people say they've got diabetes, or you've got a doctor diagnosed they've got diabetes. What sometimes is unclear, is it type 1 or type 2? The vast majority are type 2, but we do get misclassification that you can improve on it using genetics or better phenotyping. And people like Andrew Hattersley and Angus Jones have developed better algorithms, or Nish have done that, which tightens it up. It doesn't tighten up much, but it tightens up sufficiently so you get more close to people who generally got type 1 versus type 2. So, yeah.
Maybe last word for Shen. Depression diagnoses from ICD codes and read codes?
In general, I like electronic health records because it's not just diagnosis, it records the time of diagnosis. So beyond just ICD-10, people argue that it doesn't capture all depression or subtypes of depression, but I like electronic health records. One thing that really bothers us as researchers is that you have to get approval from GPs, and meaning that electronic health records are not quite available for all people in Generation Scotland. And I just hope one day that health records will be available.
GPs in the house, actually. You want to have
I'm sure they're all approved.
Privacy as well. Lots of things.
That's true.
Okay, super. With that, I think we'll say thank you ever so much to both speakers. We now have a... We now have a 30-minute coffee break, and we'll be back at 1:45.
Okay, can you hear me okay? So good afternoon and welcome to this final session. Haven't we had a great day? So we've talked a little bit about the study, its provenance, its origins. That was the first session. And fantastic couple of sessions there. Then we talked about the research productivity of Generation Scotland, which has been great. And now, thirdly, we're going to talk about context and look forward in terms of opportunities in the broader health data landscape. And we've got three terrific speakers, so I'm glad that we've had no audience attrition because they are well worth staying late to hear from. I should perhaps introduce myself. So I'm Andrew Morris. I'm Professor of Medicine and Vice Principal at the University here in Edinburgh, sort of in exile in London at the moment, doing a couple of things. But Generation Scotland is just a fantastic study. And I think, was it David referred to, I was one of the original gang of four in the early days who got it off the ground, which I thought was a great compliment, but I didn't actually know who the gang of four were. And I looked it up. And it was four Chinese gentlemen who accompanied Mao Zedong, Chairman Mao. And to coin a phrase, they used propaganda, purges and fear to maintain control and to encourage violent struggle sessions. So that's what they did. But I don't think today has been a violent struggle session. It's been a celebration. But I think it's right and proper. We look forward to the next 20 years to think how we can garner the team, the expertise, the talent, the expertise to actually drive change for the benefit of Scotland and humanity globally. And that's the feature of this session. So we've got three speakers. We've got Andrew, Cathie and Nic. But we'll kick off with Andrew. So Andrew McIntosh he's an academic psychiatrist and Professor of Biological Psychiatry at the University of Edinburgh. And he's just a wonderful chap. And he's going to talk to us about Generation Scotland's contribution to better mental health. So join me in welcoming Andrew.
Thank you very much for the introduction. I'm going to speak a bit more about some of the findings that you've heard so far. I'm going to make some light suggestions about things that we could think about in future and flag a couple of things that are quite speculative but might make future studies and might help us focus on where the eventual clinical and policy impacts from Generation Scotland could arise in mental health. I thought I'd start by saying, what do we mean by mental health and what am I going to be speaking about for the next 15 minutes? Well, mental health, broadly, is about your emotional, psychological, and social health. And the great thing about Generation Scotland is it's enriched for measures of mental health. So in Generation Scotland, you've got measures of stress response, you've got lots of measures of your thinking skills and memory, you've got lots of measures of social health, including loneliness and other factors. And these are very relevant for the health of populations and for their well-being. The flip side of mental health is, of course, mental ill health or mental illness. And by mental illness, usually what people think of first of all, they usually think of things like schizophrenia, bipolar disorder, very severe forms of depression that are admitted to hospital. They also think of common mental illnesses such as anxiety and depression. And anxiety and depression is, as many of you will know, extremely common in Generation Scotland, as it is in other studies across the UK also. And symptoms of those conditions have been growing over time for reasons that we don't fully understand. Like Naveed and like Blair and others, I'm also a clinician, and a lot of my work is in the clinic, at the Royal Edinburgh Hospital here, and treating people with depression, schizophrenia, bipolar disorder. And the treatments of those individuals hasn't advanced as fast as it has in other parts of medicine. And I think understanding the mechanisms of that are going to lead us to better treatments and to better policies that reduce the impact or reduce risk. So I'm going to speak about some of the things that I think are relevant to how this might develop in future. First of all, I'm going to remind you of what you did for the participants in the room, exactly what questions you answered in Generation Scotland, because the data collected in these is going to be available in perpetuity, more or less, and will be available to other researchers going forward. So in your baseline assessment, I'm going to have to look at the screen because the image on this here is like really small, so I'll look back here. So there were measures of bipolar disorder and of depression, and these were very rigorous measures for a population-based study, unusually rigorous and reliable measures of those conditions. There were measures of schizotypy, which is thought of as a personality trait that's related to liability to schizophrenia. There were mood disorder questions that relate to bipolarity and how susceptible you or family members might be to bipolar disorder, either now or in the future. And there were measures of things like the severity of depressive symptoms. There's a massive amount of data in Generation Scotland that is really valuable for the study of personality and cognition. I won't say much about those today, but I think of them as also falling under this broad umbrella of mental health. And hopefully you'll see other people talk about those in future, if not today. So the baseline assessment was followed up with a remote questionnaire that was sent out to many of you in 2015, 2016. And we collected further measures of depression during that time, measures of threatening experience. Some questions were quite difficult to answer, I imagine. They were they probably provoked anxiety in some people. Again, some measures of severity of depression, but we added measures of tobacco and alcohol use and misuse, and we made measurements of resilience and coping style. And then about 1,000 of you were invited to take part in a study in Aberdeen or Dundee, and you went to a brain imaging assessment. I don't know how many people in the room were part of that, but a number of you, I think, will have been. And you completed more questions about your cognition, about your personality, depression. Then we added measures of childhood experience, substance misuse, and many of you had a brain scan. About over 1,000 people had a brain scan in Generation Scotland that measured the structure of the brain, the function of the brain, and the connections between different parts of the brain. So I'm probably telling you things that you already knew. In addition, on top of this enriched data set that's been so valuable going forward, there is layered on this information electronic health records. So we have linkage to your electronic health records, and that means we can find out what happens to you and your mental health over time. And we can do it in a way that doesn't involve you being recontacted. It's what's called a participant passive method. And it's extremely valuable because hopefully everybody who's got an electronic health record in Scotland can be linked to this information. And it's got very broad reach. Births, maternity, prescribing records, something that other parts of the UK, most other parts of the UK don't have, is an extremely valuable resource. Antidepressants, for example, are some of the most prescribed drugs in medicine. Many people in here will be, I guarantee, taking antidepressants right now. And there have been other parts of Generation Scotland that have added further mental health information. So COVID Life, Teen COVID Life, and Rural COVID Life, you've heard of, also contain information that's relevant to mental health and ill health. So that's all very well. I want to tell you a bit more about what's arisen from Generation Scotland. I'm going to start in a slightly strange place, which is to talk very briefly about the economics of doing research and about how that involves winning funding and then sustaining the funding and sustaining cohorts going forward, and how what has happened in Generation Scotland so far. You'll know that it was funded at baseline, and then there was a follow-up, there was another application for funding, which was also funded, and it's enabled Next Generation Scotland. I'm not going to talk about those funding, but I want to make the point that those initial pots of funding draw in extra investments from throughout the UK and internationally that wouldn't be possible if it weren't necessary, if it wasn't for the funders' initial faith and understanding the promise of this very unique Scottish cohort. So these are the mental health specific investments that Generation Scotland has generated in addition to its original resource. So there were further mental health investments. There was a study called Straddle that was funded 4.75 million from Wellcome, an investigator award, another 2.5 million. And we currently have a grant that's called AMBER, which is looking at people who receive antidepressants and trying to understand what antidepressants do to your body, how they might work, and why some people respond better than others. Because some people don't respond well to antidepressants, and we want to find out why. So this is the near future, this grant. We've also been approached by people in a new initiative called the UKRI Mental Health Platform. The UKRI Mental Health Platform follows a lot of Generation Scotland's good principles of data sharing, of making data available to other people, of having very well-designed governance procedures so that people can access that but maintain their privacy. And that general approach has been embodied in this new thing called the UKRI Mental Health Platform. And there are two hubs named there, Immunomind, which looks at immunology, inflammation and mental health, and metabolic psychiatry that looks at the influence of diet and metabolism on mental health. These are both major UKRI investments that have also leveraged and seek to collaborate with Generation Scotland and its investigators. But you might ask, what have we learnt about mental health from Generation Scotland so far? Now, when I prepared these slides, I've sat in the back of the audience today listening to other people talk about some of the mental health findings, and I'm going to repeat some of them just now. But right at the beginning of the day, people have been talking about how Generation Scotland can respond to public health challenges. And I think probably the one at the bottom is the most obvious recent one, if one can still mention the word COVID. COVID life, COVID teen life, and rural COVID life. These were studies that looked at the pandemic impact on people's health, physical health and mental health. And Generation Scotland responded very agilely to this new threat. They look at well-being, social, work life, and education and the effects of the pandemic on those features. And the studies have been published, as I will show you. The other one above is on loneliness in the digital world. And this responds to not so much of an acute problem, but because of a growing problem, is particularly in Western cultures, where people are very worried about the influence of social media and growing loneliness, particularly in young people. And again, Generation Scotland responded extremely well to that increasing concern. But that wouldn't matter in a sense if it just appeared in a journal or it appeared in a publication and scientists read it. That would be an achievement. That would be a worthwhile thing to do. But I think what we're all motivated by in this room is of getting better treatments for people with mental illness in future, of improving the well-being of populations. And so one of the things I think that is highlighted probably too infrequently is that Generation Scotland has been able to influence policy through some of these initiatives. So these two studies here, so some of these people in the room, Alex Kwong and Nic Timpson here, David Porteous, again here, David Porteous in the bottom, publications on COVID, were presented to government. They were presented to the Strategic Advisory Group for Emergencies, or SAGE. They have been used by Public Health England, and they have been read, digested, and used by Scottish Government. So this is not just scientific research for scientific research's sake. It's scientific research that will hopefully enable and improve the health of this and future generations. So digging into some of these findings again, because I think in some of these examples, I think signpost ways in which Generation Scotland can be influential in future. Genetic discoveries in depression have accelerated at a much slower pace than they have in other areas of medicine. But now, thanks to Generation Scotland, in this study, we now have nearly 700 genetic changes throughout the genome that are associated with depression. Again, that sounds like something that might not be obviously relevant to the health of people in this and outside this room, but we're now able to train polygenic risk scores, so genetic risk scores that can predict depression with a better than chance in other ancestries, as well as in Europeans. So that is, again, an achievement and shows how this work has moved forward. When we talk about genetic changes, we're often talking about the letter changes. And the changes in your genetic code are often not the most important thing to know if what you want to do is develop therapies. You want to know the genes and the proteins that are affected. And Generation Scotland has helped to identify some of those genes for the first time. Not only that, but the genetic findings from these studies, the genes that are associated with depression are the ones that are often targeted by antidepressants. Now, that might seem, again, like an academic finding, but it suggests that we can interrogate the findings from genetic studies to find potentially disease-modifying treatments that are better than the treatments that we currently have available at the moment. You've heard already today about epigenetic discoveries in depression. You've heard about some of the epigenetic findings that are associated with the condition, but also with its risk factors. And that enables us, because the type of epigenetics we've measured in Generation Scotland is like an archive of your experience and of the things that you've been exposed to, risk factors that we know about now, risk factors that we'll discover in future. These, I think, the promise here, I think, is not only in the discoveries that have already been made, but how that can be leveraged in future when we think about how it might influence clinical care. The epigenetic risk scores that you've heard about today from Shen, Riccardo, and others could be used in future to think about how we might develop blood tests that might stratify people into types of depression, because we do not think depression is all one condition. We think it's probably many conditions. We can identify epigenetic signatures of treatments. So we can look to see whether somebody who's taking an antidepressant shows the changes in the blood that you would expect to see in somebody who took and then responded to that drug. And that has widespread application beyond mental health. But it's also helped us to identify things like inflammatory and immune pathways that are associated with depression too. The final thing I want to talk about in terms of discoveries is brain imaging. So brain imaging in Generation Scotland has been a very productive area. Many publications arising from the results in Generation Scotland, led, I think, in large part by Heather Whalley, who you heard about earlier today. So we know now that the genetic changes that are associated with depression are also associated with changes in the brain. We have yet to understand how that links together and how that leads, those genetic changes lead to changes in the brain, lead to somebody in front of you in clinic who has the condition. We also think that we are discovering now how antidepressants may also affect brain structure. These are all signposts, I think, for future studies in which Generation Scotland can play a large part, I think probably particularly in young people. A couple of slides before I finish. I want to talk now just very briefly about the value of family data in a study like Generation Scotland and what that enables you to do in terms of teasing apart things that look heritable but are actually because of your similarity with your parents from things that are genetic. So the family structure of Generation Scotland is really unusual. But it has inspired other studies that have adopted a similar approach. So perhaps unimaginatively, we called this study Generation Malawi. And you might be able to detect a saltire in the logo. But this was a study that was developed and is now 5 years in, developed in rural and urban Malawi, that follows a very similar structure to the successful structure, I think, of Generation Scotland, but is based in an African context. And you can see some of the pictures from the clinic and from the neonatal clinic. And embedded in this, we also have a birth cohort. So this, I think, is a really rich place to think about how we might make discoveries in one sample and replicate them in an African context and vice versa. Because things that are likely to be globally relevant, I expect we'll find lots of risk factors that are present in both populations. Finally, I'm going to talk about some of the future possibilities for mental health research in Generation Scotland. I'm going to talk about the thing that kind of most excites me. There are many things, I think, where Generation Scotland could play a pivotal role. One of them, I think, and I think you've heard this very briefly, is about accessing people's neonatal blood spots. I think that's a very exciting future prospect for Generation Scotland that would improve the generalisability and representation of people across Scotland, and that would, I think, have a major impact. But I want to talk about a problem that we have in some of the genetic research that we've shown here today, and that's that we have a massive global ill health burden. There's literally over 1 billion people in the world suffering from mental illness. We're developing a better understanding of it using brain imaging and genetics in Generation Scotland and in other studies, and we want to get to more precise diagnosis, and we want to get to better treatments. And what we're lacking there, I think, is the very basic lab neuroscience that's used to study these complex disorders that are present in large numbers of the population. So if we want to get a handle on that, we need to be able to find a platform in which to conduct the sort of wet lab, white coat kind of experimental studies that lead to a better understanding of mechanism at the level of the cell and tissue. And to do that, I think one of the things that we're trying to do next is develop a cell bank from people in Generation Scotland. And some of you will be invited to take part in this shortly. So this involves volunteer blood sampling of people at the moment who have been taking antidepressants. What we're aiming to do here is take a sample of your blood and treat it in a very specific way so that we can separate the cells and study them in culture in the presence and absence of antidepressants. And in future, we will be able to transform these cell lines into neural tissues. So using new technologies, we can transform these into neural cell lines in glia, the sorts of cells that go into make up your brain, and into more organized tissues. So very briefly, Generation Scotland's led to many impactful discoveries in the field of mental health. It's generated inward and external investments. It's sparked many ideas and new studies, and it's been an exemplar, I think, of excellent team working, collaboration, and data sharing. And I think it will be well positioned to respond to future health and scientific challenges. This slide is intended to thank some of the people in the room, but I think because there are so many people who've participated in Generation Scotland here, I want this to be for you. I want you to know that we really value the data and samples and time that you have given to take part in Generation Scotland, and how you continue to fill in questionnaires that have generated new insights, that have attracted young people into mental health research who otherwise would have gone to other parts of the economy, but are now working to make, I hope, the future better for people who suffer from mental illness. So I'm going to stop there, and apologies, I think I've gone a minute over time.
Terrific, Andrew. Right, and now, as the second feature of the triple act, a very good friend, Cathie Sudlow, who is the director of the Usher Institute at the University of Edinburgh and the head of the School of Population Health Sciences, with a very long pedigree of doing many great things. So, Cathie, there is no one better to talk to us about Scotland's opportunities to enable population health research and innovation at scale for the public good. So, Cathie.
Thank you. Andrew, I love the way you referred to your 2 little jobs in London with a kind of flick of the wrist. So we might out those. So Andrew is president of the Academy of Medical Sciences and also runs the National Institute for Health Data Science called Health Data Research UK. So he's quite a busy guy as well. So I want to sort of set some of the work of Generation Scotland in the broader context of what goes on across the UK in the space of health data, and perhaps highlight some of the particular opportunities, examples of good practice and particular opportunities from Scotland. So I won't be talking just about Scotland, but you will notice a few particular Scottish examples in what I have to say. So I have been lucky enough in the last few years to work with Andrew and others in Health Data Research UK. And during my time there, I was able to work with others to help progress the UK and the UK's health data towards this vision of being able to address big health challenges in the context of the entire population of the UK. Some disorders are rare and can't be studied without access to a very large population. And for some types of studies, many actually, you really want to be including as many people as possible from very diverse backgrounds, from across a broad geography, and we're better in many respects for many of those studies than the UK. We have a large population on a single landmass. Most of our healthcare is provided through the NHS, which isn't quite a single healthcare provider, but at least it's a kind of common system. So there are some particular structural advantages in the UK. And I was also lucky enough, well, I'm not sure lucky is quite the right word, but anyway, I was privileged to be invited in the last 18 months or so of my time at Health Data Research UK by some colleagues in UK government to produce a review of the UK's health data landscape and make some recommendations for progress in that field. So here I'm acknowledging some of the structures that provided me with the support that was necessary to undertake that quite chunky task. So some of the key points from conducting that review, which went on for about 18 months, and it was published in November last year. So it started under a previous government and landed under the new government, but had been commissioned by those without any particular political party affiliation, which was helpful, were perhaps not too surprising, but there was some kind of fairly resounding messages. So the first is pretty obvious, that we use health data every day as health professionals. So I, like many colleagues here have life as a clinician, a practicing doctor, as well as in science and being a data geek and all of those other things. We use health data all the time, safely, to improve people's lives. We need to collect data about individuals. We need to retain it over time and refer back to it, both for particular individuals and for groups of individuals, to be able to do that job properly. And by and large, I think we use it well and responsibly. And people support us as health professionals in doing it, and they generally support researchers to use their data, not just to benefit themselves, but other people. And many surveys over time have shown that. There are, of course, those who are not so comfortable with that, and there are dissenters. And also, even amongst those who are super enthusiastic, there are some caveats and some conditions. They want to know what's going on. They want to be told about how their data are being used to improve their own care and the care of others. And they want to know who's using it. So these all seem completely reasonable. And they want to know who might be making a profit out of it as well, and they may have something to say about that. But by and large, people are very supportive. And we have very abundant sources of health data in the UK. They come from the healthcare system, of course, but they come from way beyond the healthcare system as well, data sets relevant to our health. And one very key important point is that single data sets or single sources of data can be hugely informative about our health and provide us with insights about our health. But the real magic happens when different sources of data are brought together for groups of individuals through using processes that we generally refer to as linkage, but it's essentially data fusion exercises. It's when you bring together these different sources of data from primary care and secondary care, from death certificates and the education sector, for example, that you really start to develop the most informative types of insights. So that linking of different sources, critical. But those of us who have navigated this space for a long time, or who have tried to overcome the challenges of trying to access those data to ask the types of questions that we know is in the interest of the public, on behalf often of public funding agencies or at the ask of members of the public, find that there are lots of barriers and obstacles to being able to access and link and pull together these different data sets to provide these analyses for public benefit. And these barriers largely arise from the fact that we've got very complex, difficult to navigate, and duplicative, inefficient systems for managing and accessing data. And one of the things I've been very keen to push hard is that if you have a very complex system, it's actually easy for it to break down and for errors to be made. So the more straightforward, the more simple, and the more streamlined we can make our systems, the easier it will be to make them safe and secure, to protect everybody's privacy and security as well as realize the benefits. So we need to get rid of the complexity and the duplicity in our systems to enable that. Because this is preventing and delaying crucial insights that are of relevance to millions of people who fall sick and become ill from a multitude of conditions. So this is about everybody, and in not doing this well or well enough, we're letting people and their families down. So the UK's health data should, in my view, and I think in the view of many others, be regarded as part of our critical national infrastructure. And luckily government, I think, have really taken that on board. So here's the Office for Life Sciences summary of the review work, and it took me about 60,000 words to say what I had to say, and it took me 18 months, and they slapped it all onto one slide, and I think they've done a really great job, actually. So this is a great takeaway message, that we've got fantastic sources of data. We're not making enough of them currently. There is huge untapped potential for a range of different uses, and you can see some of them, the major ones, summarized on that slide, from the impact in healthcare right through to research and innovation and new development of new insights for new drugs and new therapies and new prevention strategies and so on. But we can only do that if we have a proper joined up strategy that involves a number of key steps, and I'll come on to those again a bit later. So what are the sources of data across the UK? So here on the top half of the slide, you can see sources of data that are collected every day about all of us in our interactions with healthcare and other public-facing systems. And some of those data sets are brought together at national whole country level, and increasingly in particular during the pandemic, when England caught up and Northern Ireland caught up with what was already going on in Scotland and Wales, these national assets, data assets, tend to be brought together in safe, secure settings where data is not shipped out to researchers to generate their insights, which wouldn't be as safe and secure as it could be. But researchers come to the data, which is held securely with all the identities of the individuals who it represents stripped away to make sure that it can be looked at and analysed safely and securely with approval mechanisms both for the projects that those researchers are doing and the way in which they are interacting with the data. So these are called secure data environments or safe havens or trusted research environments. And then we also have, in a number of regional and local settings, much more deep and granular data, which increasingly can now be brought together as well at whole country or national scale, provided we have the right kind of access mechanisms. We don't necessarily need to pull the data into a big data lake, but we need to make sure we know where it is and know how to pull it together for particular analyses. So these more granular and deep and complex data involve, for example, data from images. Here are images of our brains, but you could have images of all sorts of bits of people. All of the kind of free-standing text in correspondence in medical records, and the laboratory data that's generated in labs up and down the land every day in NHS and other clinics. And then also relevant to our health are the data that are gathered about us and about our families in other settings, for example, education or disability data sets, income data. We know that poverty is very closely connected to health, for example. We know that educational opportunities impact later life health and indeed vice versa. And then there are other sources of data as well. Our own personal mobile phones, wearables and club cards and things collect data about us, and with our consent, those can also be linked to other sources of data to develop insights. There's some great examples of that. There's data in the environment from weather monitoring and air pollution monitoring. And then there's research-generated data. We've been talking a lot about that today. So research studies like Generation Scotland or other studies which I've had the privilege of being involved in, UK Biobank or Our Future Health, which is now firmly arrived in Scotland, and many of you may be participating in that. Large so-called observational studies that often collect samples and all sorts of other information from people. And then clinical trials like the recovery trial, which many of you will remember, ran very successfully during the pandemic and provided a lot of information on treatments that did or did not work for the treatment of people with COVID in hospital. So it's by linking those sources of data to all of these other sources of data, the day-to-day administrative collections of data, that we can really realise the benefits of these large, major and very expensive research studies and make sure that we milk them for all they're worth. So here are some examples. This is one from medicines data. So there used to be a collection of medicines data only in Scotland for community prescribing from general practice. But now actually Wales and England have caught up. So this is a 3-nation study looking at medicines prescribing and dispensing during the pandemic through a cardiovascular prevention focus. So you can see here these big dips early on in the pandemic when we had lockdown of people picking up medicines from pharmacies for classical medicines that are used to prevent strokes and heart attacks. And as a result of that, because people weren't getting out because of lockdown and they weren't getting to their GP and they weren't getting their prescriptions, we reckon that about 500,000 fewer people than might have been picking up these medicines were picking them up and taking them. And probably there was an excess of heart attacks and strokes as a result of that. So that was a very interesting study, but also one of the first to do a study in over 60 million people, drawing from data across all of these different populations. And then as a follow-up, and a lot of this work was led by my colleague Xueyi Shen from the University of Liverpool, there have been further similar studies looking at anti-epileptic treatments and the change in the use of these anti-epileptics during the course of the pandemic and the impact across society on different groups of individuals. So fantastic for medicines monitoring. Unfortunately, these types of studies are not very easy to do, and they are not in any sense business as usual. To my mind, the Medicines and Health, the MHRA and other medicines monitoring organisations that have an interest in the safety of medicines should be able to be doing this at the touch of a button. This is the sort of information that we should be serving up to allow us to monitor medicine safety. We'd have far less disasters as a result. There are other studies that have been done, again, as a result of advances made during the pandemic. Here's an example about drew from data sets across all 4 nations of the UK, looking at the under-vaccination, so people not being vaccinated as they should and as was recommended against COVID. And you can see quite alarmingly high rates of under-vaccination by the middle of summer 2022. So initial high uptake was followed by a decline, and there are, of course, quite important consequences of that. So there are some great examples of these more granular type of data assets around the UK. And one in particular, which I think makes me feel very proud to be up here in Scotland, is the Scottish Medical Imaging Archive. So Scotland's NHS radiology is gathered on one common system, and it's provided a huge opportunity to link the information from those images and analyses of those images to other parts of the health record. And because they go back quite a long period of time, over 10 years, it's allowing people to start to build models of how changes in brain images at very large scale can predict later life health. For example, heart attacks, or importantly, dementia, which we know we need to predict early because we know that to prevent it, we need to treat people early. So we need these mechanisms. And colleagues up here in Scotland and elsewhere around the UK and internationally are working very hard on data sets like these. But I couldn't emphasize enough, this is a really globally fantastic resource. There is nothing like it anywhere else in the world, and it's here on our doorstep in Scotland. We also have the capability to link data from different sectors, so from the NHS to other sectors that are relevant to our health. So Wales has been particularly good at this, and you can see here a Welsh example of school absence and the influence that has on later life health in young adults. But also England has been catching up and starting to link data from the health sector with the education sector. We have this capacity here in Scotland as well, and Roger Halliday's leadership of Research Data Scotland is really helping there. And here's one example of how it's been possible to identify people in care homes with dementia, which was obviously something that was incredibly important during the pandemic, to know who was in a care home and what was wrong with them. And that has been done by linking together data from the social care sector with data from the NHS. But importantly, this is work that was first aired at a conference in 2023, but it's using data from nearly 10 years previously, which I think illustrates very nicely how long it can take to be able to access data, to be able to do these types of studies. And we should, in my view, be doing this much, much faster and much better. We can also use these types of routinely collected data from health and other administrative systems to enhance and improve the efficiency of large-scale clinical trials. So the recovery trial I've spoken about previously, there's a fantastic trial that the NHS has been running across the country, mainly in England, using centralised services to invite millions of people to take part in a trial of cancer screening tests. And closer to home, the aspiring trial of brain hemorrhage, a subtype of stroke, has used national data both to invite people, to find the right people to invite to take part in a trial of treatments for brain hemorrhage to prevent future disasters from occurring in those individuals, but also for following their health over time without having to invite them back to clinic, which is obviously a much more expensive way of tracking them. So we also have, of course, these large population health cohort studies, like Generation Scotland and many others, that use linked health data to follow participants up over time at huge scale. This is incredibly important. We'll hear a bit more about some of these from Nic shortly. But UK Biobank is one of the largest and longest established of these studies. We heard a bit about that. Generation Scotland, you've heard lots about today, and that has grown fantastically, both in size but also in scope in terms of its extension to younger individuals. There's Our Future Health as well, which is now recruiting at even larger scale across the whole of the UK, with a fantastic front door for our colleagues from industry to be able to come and run experiments that help us to find new early ways to diagnose and prevent disease. And then a newer one, which I'm privileged to be helping to kickstart, is the Adolescent Health Study, which is aiming to recruit young people across the UK to try and provide further insights into that very important point in life where children go through teenage and grow up to be young adults, and lots of things change, and there are lots of challenges that need evidence to be brought to bear. So we can only do that if we can link the information that we collect directly from those participants to their health and other administrative records. So there are lots of ways in which data can be used, and I've shown you some great examples of what's possible to save and improve people's lives and to support research and innovation. But actually, these success stories are all too often the exceptions. There are lots of ways in which this is complex, there's too much friction, there are too many delays, and there are lots of gaps. We could be doing it a lot better. And so my review made some recommendations. I made the point about critical national infrastructure. I suggested that we should have a national health data service to allow us to really take this forward with proper investment and proper high-level accountable leadership to really put it on the front page of the government's agenda. With, of course, the need for ongoing leadership and engagement from patients, the public, and importantly also health professionals and others, with a number of UK-wide approaches to standardise and streamline everything that we do at whole country level. And somewhat surprisingly, because quite a lot of reviews that are written for government sort of have a brief period where you hear a bit about them and then they sort of go quiet and they sit on the shelf and gather dust. This does seem to be having an impact. It's informed some of the plans that we've seen coming out from UK government as well, and it seems to align with the government's agenda around the good use of data for public benefit. And the Prime Minister in April 2025 announced that there would be major investment in a new health data research service. So this felt like a good direction of travel. There's everything to play for now because this new health data research service will soon be being set up. There's been a little bit of doldrums and planning blight while we've waited for this moment. So there's a lot of hope. It has some key features. It will be a UK-wide endeavour. And so there's everything for us to play for in the devolved administrations as well as across the whole of the UK. There are some core kind of whole population national data sets that need to be key pillars of that. We heard about the importance of GP data. It can't be overemphasized enough. But there are some also important underpinning principles. And in terms of opportunities for Scotland, I think it's really important in Scotland that we continue to take a one-nation approach, that we join forces with some of our regional data assets so that they can be combined onto at least one virtual platform, because fragmentation and duplication is not sustainable, it's not affordable. GP data, we do need to have routes to access that at national scale. It's so important for all those conditions that are managed outside of hospital. Things need to be properly aligned, and interoperable is one of the words that we use a lot. And we need to learn to share. So organisations do not all need to build their own fiefdoms. They need to learn to build on top of each other's fiefdoms and combine forces. It's much more efficient, it's much more effective. It's kind of a no-brainer for members of the public, but for people who like to run stuff, they always want to be in charge of their own thing. It's something to be resisted. In Scotland, we've got some fantastic key assets, and we need to recognise them for what they are. So Scottish records go back a long way. We have fantastic longitudinality of records and of expertise. We've been doing this data linkage thing for longer in Scotland than any other nation in the UK. We have a fantastic company which can help us with access to and linkage of GP data, which has been recently acquired by National Services Scotland, so that's a great opportunity. We have the Scottish medical imaging resources I told you about. We have fantastic medicines data in this country. It's better than any other part of the UK, so a fantastic test bed for safety studies and many others besides. And you heard a bit about the neonatal blood spot archive. So it's not yet used for research. It has fantastic potential, and I know that Dame Anna and many of her colleagues in Scottish Government are working hard to start to lead us towards this being a reality in the future for Scotland and beyond. So I think some of the biggest blockers that we need to recognise and address are a tendency to complacency. Because we've been doing a lot of this stuff in Scotland for a long time, we think we have it all taped, but actually other countries are catching up, and it's on us in Scotland to continue to experiment and lead the way and show how things can be done so that those can be shared with other nations across the UK. And we also have a tendency to think a lot about the risks and the harms of uses and sharing of data, particularly in our big bureaucratic bodies, and we sometimes forget about the benefits. So if you think about crossing the road, you cross the road so you can go and get your stuff from the shop on the other side. There's probably a 1 in a million risk you might get knocked down on the way across. But the benefits of getting across the road to the other side outweigh the risks. And I think sometimes we've got that risk-benefit equation the wrong way up when it comes to uses of health data. So we need to be cautious, but we need to get on, use it, and use it well. Thank you.
Fantastic. And we're not going to have a panel discussion yet, actually, because I think Nic's here. Is he? Great. So the last part of the trio is Nic Timpson, a very good friend. He's a fly fisherman, he says. I don't think he's ever caught anything. He can maybe give us the ground truth. But in his daytime hours, he's Professor of Genetic Epidemiology at University of Bristol. He's a good friend of Scotland, and he runs a fantastic study called the ALSPAC study, which he may touch upon. So welcome, Nic.
Okay, thanks everybody. Thanks for having me along. Like every good and well-positioned bookend, I'll be as swift and sturdy as possible and get off the stage so you can have a discussion. And, you know, as an imposter, the sort of Englishman in the corner, I've been surrounded and been stared at by Robert Wallace and other eminent scientists who have just made me feel inadequate and useless here. So I'll just keep quiet and get on with the main points. I think my job here really in this last session, just before the panel set, is to give that focus of what GenScot is and where it sits in a broader landscape. And I do that with my position as a cohort PI myself. So I look after a cohort in the southwest, but also in a position of trying to do things in a way that enables connectivity and infrastructure across cohorts. And I've been really, I've really taken home some of the points that have been made today, thinking about some of the, the desire to translate findings within our own cohorts from our participants into something real. I think Blair made that point earlier today. Also thinking about some of the points during Naveed's talk about that, so what for record linkage? Sometimes the administration and the data aren't as good as we want, but we have Generation Scotland with perfect data that we can calibrate those against and make better for many millions of people. So it's those assets and those touch points that I'll try and draw on. So my name's Nic Timpson, and has been said, I'm a genetic epidemiologist, so actually a long-standing friend of Generation Scotland and many in the room in that area. But I'm going to talk today in a more broad sense about cohorts and their value. We've seen this time and time again. We haven't had this debate before. In 2014, the MRC was sporting, actually it was only a collection of 30 cohorts, but championing the idea that these longitudinal collections of volunteer data and biosamples are useful. Again, in 2019, the Wellcome Trust LPS Longitudinal Population Studies effort was to get behind these studies with infrastructure and keep them moving along. And we've heard a lot about COVID today. Sadly, one of my examples is COVID, but I'll be quick on that one. We were trying to draw upon what is the collective value that we get from population-based collections and study repositories in order to make things better. Well, it's about variation. Very rarely in the world of epidemiology do we ever have anything that's truly representative in terms of a collection or a sample. What we do have is pockets of incredible depth and then administrative records that sit across them with which we can make really good contributions to evidence and data. And it's that variation we need to keep going and keep our participants animated by. And indeed, most recently with the industrial strategy, it's not much space, but we have a line in there which is championing cohorts and longitudinal data, the likes of which really aren't found anywhere else in the world, bar some of the Scandinavian linkage efforts. We really do it incredibly well in the UK. This is my study. It's called Children of the 90s. You might have heard it called ALSPAC today, the Avon Longitudinal Study of Parents and Children. Lots of exciting findings. I present this slide quite often. However, we're talking about Generation Scotland today, and it took me less than 2 minutes to find the equivalent slide. Fabulous science repeatedly coming out of this wonderful study, and also a depth which was really exciting, really good outreach champions of your science, reaching out to the populations and making policy differences. That's really exciting, not just, I think, for participants, but it gets the researchers going too. What's the value of my effort if it only stops at a manuscript? What I'd really like to do is address a challenge and make a change to the broader population, or indeed further than my city or my region. And you see that in spades with Generation Scotland. What we can do, I think, is maybe even more than one region. We can think out of the box a bit and about connectivity. And why is it exciting to think about bringing population-based assets and cohorts together? I think there's a number of reasons why it's exciting. Ironically, we think about longitudinal cohorts as slow and laborious, expensive exercises. However, contrast that with the challenge of starting a study tomorrow, which had to address a policy question. If you've got one that's already going prospectively, generating wonderful data and evidence already, but you can then reach out to it immediately to address a problem, local authority need, a national need for that matter, then it can be incredibly responsive and agile, just as we've seen for some of the examples. Within those collections, we also have variety. We have representation of different parts of the UK, different people, different ages, different ethnicities, different biologies, and clinical procedures and presentations. They're very deeply characterized. They've been followed often through time, and that gives us an angle on the emergence of health and disease, which we wouldn't get in cross-sectional studies or trials for that matter. We have an ongoing working relationship with our participants. It's a two-way enterprise. In the southwest where I am, we're afforded the luxury of being a city-based study, and we have an intimate relationship with our participants. We've been running for 30 years, and the reason that we still have half of the people left who were involved in the beginning is because we talk to them all the time. We're listening and receiving evidence, we're co-designing and moving the study along together. And indeed, it starts to grow when you wait that long, because you start to get children of your children of your children, and it goes on. We get data and biological samples from those participants, which we're able to base our research on, which you've heard all about today. And I think really a key point, and I'll raise it right at the end, is that they leave a legacy, a longitudinal legacy about the public's involvement in science and steering policy, which is a positive interaction rather than just one of command and response. I won't dwell on these because you've heard some of these examples today, but they're nice for me because they are my study and Generation Scotland working together. So in COVID, we had that happening. The bit that I will focus on is and there's no arrow, unfortunately, but if I could leap or had long arms. Early in the pandemic, 9th of April to the 17th of May 2020, a questionnaire was deployed and data were gained. That's fast. I mean, that's really fast to bring together a data collection exercise across separate parts of the country with coordinated assets, which then led straight to policy-facing information. Really exciting. Sad news in so much as there were age gradients about the impact on anxiety and depression because of lockdown, but important evidence. And indeed, because of the nature of the studies, their granularity, one could then dissect those features and say, well, what's leading the associations? Is it your age or your sex or your clinical status or something else? So that's to do with this interplay between the depth and granularity of a cohort and the speed at which it could move to address a problem. Super exciting. And this is two studies, totally independent, working together at the same time. And I think this is really exciting in terms of where we're sort of being pushed in terms of government direction. Some of you may have heard of this mission-led focus on policy. If you dissect that and start to look at some of if you like, the directions of government as they are now, which is important because it steers public purse, I think we can speak to these things. And it's not just building on the NHS side of things, the health and disease and so on. You look right across the missions, and I think we can add value through longitudinal studies, just like Generation Scotland. Thinking about economic growth, well, a byproduct of keeping a healthy population is keeping people in working age and going back, contributing to the economy, providing those assets for growth. Think about breaking down barriers to opportunity, the interplay between the longitudinal arrival of health and disease and getting access to education, getting into jobs, getting into lifestyle patterns which afford us to put our population on trajectories of health rather than trajectories of ill health. So I think these longitudinal studies that we have, these incredible assets, cut across the direction of travel for current government policies, certainly in England and more broadly in the UK. A quick zoom in on my study, I call it Ausbach, is Children of the 90s. Why is it a gem in the middle? I think we could put any study in the middle here. I'm just going to tell the story of the one I know. Locally, we have longitudinal data, we have regular prospective collection, we talk to our community and speak to policy-relevant questions. We connect the study, just as we've heard from Cathie a second ago, to administrative data across many domains, not just health, but the Avon and Somerset constabulary. We have access to education data. We're already record linking to transactional databases through Tesco Club Cards and Boots. Now, done in the correct governance and co-developed with participants in a safe environment, we can use those data to get a better handle on what's important for health and well-being in the population. We're not alone. We're part of a community. We've heard about some of these resources. There are the giant studies like Biobank, Genomics England, Our Future Health, alongside record linkage, and then a swathe of other studies, cohorts like our own, which are being connected by other, if you like, meta-agencies, Administrative Data UK, Health Data Research UK, and so on. So there is an incredible environment in the UK which is providing that infrastructure and connection for studies in order to do better research. And lastly, I think there are initiatives which bring together regions and are making this, if you like, scaled up operation. In the Southwest, we have this, in fact, if you think about the integrated care boards, so the way in which our trusts, our healthcare trusts are organized in a region, they're starting to provide to get data together in a combined and coordinated way in secure data spaces that promote disease research. And we're not alone in the Southwest. This is happening all over the country. Quick zoom in on what's happening and why the UK is particularly special in terms of studies that are already out there. Well, Louise Arsenault at KCL was charged by the Wellcome Trust for bringing together an overview, a summary of available longitudinal studies, not just in the UK, but actually internationally. Now, this work has brought together a thing called the Atlas, which gives you a relatively light touch feel for what studies are already available and collects some information about them. If you mind that, you can look across the 3,000 studies that are out there internationally, and you can pull some details about what's available. And if you think about the ratio of population size to the number of studies that are out there, the UK starts to hit a bit of a sweet spot. Not only do we have many assets, we have a relatively small population size, and we start to get coverage that's useful in terms of bringing data together. What do we have there? We're just focusing on birth cohorts. There's about 40 birth cohorts. Many of them are relatively large, and many of them have got biosamples and data connections available, and they're also varied. They've got different people involved in them from different demographics and parts of the population. Sadly, of the 282 or so in the UK, only about 70 of them have got access routes which are already pre-formed. Most of them you have to reach out in manual form to their study coordinators and PIs to try and work with them. Worse still, about 50 you can't get at all. So in some areas we're great, and in other areas we're missing an opportunity. We have this family of studies within the UK that we're not knitting together as effectively as we could do. And indeed, I would argue that there is a landscape that we could optimize. Studies around the edge, services providing the glue to connect studies together, and then these meta-entities which are looking to bring all of those data into secure places to allow analysis. I'll give one or two slides on this initiative. This is a UKRI initiative called Population Research UK, whose job it is to try and identify some of those synergies, and in the absence of infrastructure to coordinate, try and help build some of that. This is the team that lead that, but just to focus in on our objectives, we want to coordinate and help tell the story of studies to those who are in different stakeholder families, policy, industry, and so on. We're looking to enhance the ability to find studies, streamline processes to access, or at least provide roadmaps to access to those studies, think about facilitating administrative data linkage, as we heard about a moment ago, and then build on training and capacity to allow the new wave of researchers to get their teeth into these data. Now, I'll flash up a large table of objectives, but we don't need to draw upon that. The idea here is about connecting the assets that we have in front of us, just like Generation Scotland, for the better of research in the UK. If there's one thing to look at, it's the left-hand column on activity, where we're looking at community engagement, policy. We're looking at searchable registers of studies, enhancing linkage, getting studies into secure data environments, and facilitating training for new researchers. Last slide, then, why is this important? Well, a number of reasons. We obviously, and we've heard from Naveed and others, that there are a swathe of persistent health and well-being challenges. We know that. There's an absolutely upfront and central role of the public and participants in science and where it's going, And Cohort, just like Generation Scotland, are a real vehicle into making that connection real, rather than just thinking about metadata in every single person they might not see or understand. With them, and in combination with data assets that are more broad, we can genuinely, we've shown multiple times, you can make a change to policy and change people's lives for the better. And indeed, that sits in the context of new and exciting interventions, so actual vehicles of doing something better. I'm from a biomedical community, so I'm thinking about things like weight management services, mental health, and so on. But this doesn't just sit in health, it sits across opportunities, schooling, and in other areas. The data landscape's changing, so the idea of your data being shared in safe places, I think, is becoming more acceptable to everybody. We understand it better, and where it's done openly as opposed to some of the commercial sectors where we don't see what happens. Funding and sustainability is a challenge, but we need to recognise it and show off our studies like Generation Scotland to make sure everybody understands how valuable they are and what we'd miss without them. So I think all of those points taken together point directly towards studies like Generation Scotland. It's why we need Generation Scotland, to be able to tell that story, show the science, and fly the flag. Ideally, that allows us to promote the case of longitudinal birth cohorts and longitudinal studies. And indeed, I think it allows us to speak to other communities. So we all understand cohorts because we're in them or help to look after them. But where are the other communities in this room? The social scientists, the artists, the industrial partners, and so on. We need to be able to communicate outwardly. So the last slide for me is this one, which is a thank you to genuine thank you to GenScot for flying the flag and being outrageously good in everything you do. I mean, the stories that we heard this morning from Heather and others about recruitment and keeping people involved were just so inspiring. And just to say, I could have pulled any examples, but I pulled this picture from the website because I think it really speaks to the mission here, which is getting into classrooms and getting into the minds and lives of those who are coming up and taking over. And I think I go back to the discussion earlier today, complicated discussion about polygenic risk scores. How do you incorporate genetics into the way we think? Well, it shouldn't be a bad thing. But just as much as you go to a doctor and see them with a stethoscope, you're not terrified of it. It's part of who we are. So we should be knocking down those walls and educating those who are part of our studies. And GenScot does that plus. So thanks for the invite. Thanks to Generation Scotland. I look forward to the panel discussion.
Okay, panel, please join us. I think we've got a few minutes for discussion. I'll go to Robin. 8 minutes. 3 terrific talks. I'm sure it'll stimulate a lot of questions. Nic has challenged us to knock down those walls. So, any questions from the audience? Wow, we've got loads of questions. And because of time, please keep the questions really tight and also answers, because I want to get round as many as possible. Gentleman there, and say who you are, because we want to know who's in the audience. Yeah, gentleman there. Gentleman with the beard. And then Naveed.
Thanks very much. My name is Gareth Morgan. I'm just a participant in the Generation Scotland panel. I have an ethical question. Surely all medical research should be fundamentally directed to reducing human suffering, to treat illness and other conditions and so on. But isn't the outcome of these kinds of studies likely to be just increased longevity, which will then lead to greater suffering and that long deterioration at the end of life?
Okay, lifespan versus health span. What a great question.
Yeah, I think it's a brilliant challenge, and we should take it on completely. So our aim should be not just to lengthen life, but to lengthen quality life. And so I think, and there are very interesting kind of trade-offs, aren't there, in terms of, so you can lengthen somebody's life by saving them from having one illness, only to preserve them to survive to have another. And we do need to take those into account. I think increasingly that is happening, and researchers are starting to grapple also with concepts of multimorbidity, where actually treating one condition or thinking about one condition in a silo is not good enough anymore. We need to think about the fact that individuals often have a multitude of health issues, and they should all be factored in together. So it's a brilliant challenge. I think it is one that the research community has an appetite to grapple with and is starting to grapple with. Nic?
Great question. That was an easy one.
I would only add one tiny detail, which is the longitudinal component of these amazing studies isn't just into old age. It's also looking back across the life course and understanding how one got to either a position of health or ill health. So that longitudinal component is super key, and you don't see it outside the cohorts.
Are you convinced? Encouraging, okay. B+, well done. Next, gentleman here.
Thanks very much. I'm Stephen Powell and I work for Datalock here at the Ulster Institute. And if I get in a very shameless plug for us, we're the Loading Research Safe Haven, and we have very rich, deep, granular data for about 1.2 million people in the region, including lots and lots of GP data, which I understand a lot of people are interested in, and lots of community and inpatient prescribing, track data, and we also have care home resident flags and things like that. All rich stuff that you could easily work with. The kind of really quick question is basically, can we do more to promote collaboration between the likes of Generation Scotland and some of these regional safe havens and their English equivalents? And the other part is, where do you see social care data fitting within that landscape? Probably very much for Cathie, because it's an area that we're working very hard to try and integrate that data from this level within the health and social care partnerships and care homes.
Great question, and a resource we need to partner with. Cathie.
Yeah, well, I think the short answer is yes and yes. And I think so you know this because I know that the people who lead and work within the regional safe havens across Scotland that are collecting this granular data are working more and more closely together to create that kind of one nation approach. So Scotland needs to be demonstrating how that can be done. Because as Nic said, we're starting to get now this fantastic set of regional setups across England as well, and they're going to have to do exactly the same thing. So, because if you have a cohort of people in a country, they won't all be in one region. They'll be scattered about, and if they didn't start off scattered about, they'll move about because we are mobile, particularly in youth. So we need to be able to join these resources up together to operate through a kind of single front door. And I think you know better than I do that this is really happening in Scotland now. And I'm delighted to see that one nation approach is bringing this granular data up a level onto the national scale.
Okay, so we'll go to Anna and then Naveed.
So to the panel, so it's not all to Cathie, but probably to Cathie mostly. When I speak to colleagues who do commercial or investigator-led clinical trials, despite all you've described, the information governance is still a problem. And in your list of blockers, I maybe haven't quite spotted it. I think if Scotland, UK, the world is to serve patients the best way we can, we need Scottish IG, UK IG, and we're not yet there. How the panel will get me there, because I need to be there now.
Okay, how are you going to get Emma?
I'd like to invite Professor Morris to comment on that. I mean, this is about streamlined governance, and I think it's one area that Health Data Research UK has really started to add fantastic value to by drawing together the people who are leading on this across the UK to have a consistent approach. So we've got a long way to go, but Andrew.
Yeah, so we have to credit Graham Laurie with the phrase proportionate governance, because it needs to be proportionate. Secondly, people use information governance as a smokescreen to avoid change. Me, instead of us. So I think it's about being clear on the governance infrastructure. And it's both bottom up, but also top down in terms of, because at the end of the day, we are all recipients of the public pound and we're working for the public goods. So aligning the incentives around a streamlined governance infrastructure is so important. Otherwise, none of this will come to fruition. That's why we need the social scientists, the lawyers. How many lawyers in the room? Oh, welcome. We're about to offer you a job, Gareth. But this is why this is a truly collaborative effort, because that is the biggest bottleneck. And yet we haven't showcased innovation in governance today. Andrew and Nic.
You go.
I'm just very briefly going to give an impression of a meeting I went to in London a few months ago, and it was NIHR organised. So this is National Institute of Health Research. It's mostly England. There are some parts that are in Scotland, but most of the investment in biomedical research centres is in England. And they have impressively dealt with many of the challenges that are out there for clinical trials, many of the governance issues, many of the contracting issues that often bedevil setting up trials in the first place. And I suppose this is just a comment to say, I'd just like to see more of that working with England and Wales and working with NIHR, because people are kind of treading the same path all the time. And I'd just like to see more collaboration with them, and some of that excellent work move to Scotland.
Okay, happy? Very quickly, because we're on the clock.
Sorry, 1 second. So that's what we're doing. You might be aware that the so-called VPAG investment program, the CRDCs, the launch is on the 1st of September. You're going to hear it's 4 nations activity, 4 CSAs, health together, made it absolutely 4 nations, and you'll see a lot of joint work across England and 3 devolved nations, absolutely as one, with ministerial support.
Fantastic. There we go. It's like a hustings. Naveed? Yeah, just shout.
Yeah, no, so I love those talks. Connectivity is everything. Some of the questions you asked, it's a very positive question. I guess the key question to all of you, actually, with all this connectivity, we can ask so many bloody questions, but not all of them are going to be that important to changing outcomes. So how do we prioritize the questions that really will make a difference in the multimorbidity, the mental ill health in young people, the rising obesity, and all these aspects that really are the big ticket items that we need to change? I'll just give you one other thought. By 2040, 40% of our nation are going to be over 64 years of age, which is a good thing, but also I think that creates, and there's much more obesity. So those things that are aligning to give us major problems, and we're seeing it even now in the NHS. How do we align to the big, big questions and not just fixate on things that we can answer but change nothing?
Start with Andrew, and then Nick.
So again, I want to describe an experience of something that has kind of changed the way I think about this, and that has been the involvement of the patients and the public in developing research questions. If you want to find out what matters to people, what matters to the public, then you have to ask the public, and you have to ask people who are affected. In our case, mental illness. And I think this has been built into many of the new initiatives. The new mental health platform, for example, is driven with a board of people who represent the interests of people affected by the conditions. And I think that's been, until very recently, really underplayed, and we haven't recognized the importance of that. So I just really want to give a shout out to that aspect of this. I think your question has a very complicated answer. I suppose it develops on interest, on economic imperatives and many things, but I think it has neglected the view of the patient previously, and I think that's being addressed now.
Okay, I'm keen to get as many questions.
Just add one thing, which is partnership and low ego. So people being willing to work in partnership towards common goals with, researchers, public, patients, everyone in it together, but not like playing to their own personal agenda.
And final question from, if we quickly get the mic.
Training program in precision medicine that uses a lot of data, and it's great to hear of all these new exciting better governance, etc. But historically these platforms, these cohorts, really expensive to access, really slow. PhD students 2 years in have still not got their access. It's fine if you're Andrew's PhD student, but is that going to improve?
So let me speak that directly. So number one, thank you for saying that. We hear this routinely. It's exactly what we're trying to focus on, getting over that curve and helping that access point firstly, finding things and understanding the routes to data before one can then change and just steamroller access and governance, which is tough to change. Second point, though, is to say that question again and again whilst we've got the camera looking at this and it's being repeated. Funders are pulling away from good PhD programs. We want both streams. We want the finessing of access and the availability of data, which we can see is the direction of travel with secure data infrastructure that's being generated. But simultaneously, with this gift of a training opportunity to set off the next generation, we're stifling and not providing training. It's outrageous. So we need both at the same time. So a call for what we're doing now, but then a call for supporting that route to navigate through training through to PhD.
Okay, time. This great panel discussion, wasn't it? Can we thank our three distinguished panelists? So I had 5 minutes of reflections, but I don't think we've got time for that, actually. So I'm looking to Robin. Yeah, so I'll do that. I'll do 30 seconds. I was going to ask you a question. If we were here in 20 years' time, what's the one thing you would be saying wow about? There we go. That's a good question, isn't it?
I very much hope we will have a stronger notion about the causes of diseases that take many years to generate, and dementia is one of the the challenges of our time. And we will start to have some early treatments that actually make a difference to whether people develop dementia or not.
Great. Andrew, 20 years' time, what are you doing now about?
So I think I said it in my presentation. I think we know relatively little about things that affect very large parts of the population, especially in mental health. I hope that in 20 years' time what we'll have is a much better understanding of the mechanisms of those conditions through studies like Generation Scotland and things that come after it, by integrating all of the things you've heard about, but also working more with people doing basic neuroscience.
Fish will you have caught, Nick?
Oh, just one, but it would be enormous. No, the thing I would love to feel is that there is an appropriate level of understanding in the general population about data and components of it, so that this wheel can be oiled.
Okay, convergence of care, research, benefit to the population. Okay, right. So I won't say my remarks, but can I just say a few things? Anna, David, Riccardo, Roger, Heather, Blair, Xueyi, Naveed, Andrew, Cathie, and Nic have been absolutely terrific today. Can we give them a round of applause? My three key messages, which I was firstly, Generation Scotland was prescient. If you look at the investments since in our future health in UK Biobank, Generation Scotland led the way. Secondly, it was about collaboration. The participants, wow, thank you. But also it's about the social scientists, the lawyers, the behavioral scientists, the environmental scientists. The biomedical scientists should resist being elitist and actually work in partnership, both geographically and in an interdisciplinary way. So I think that's important. And thirdly, the importance of infrastructure, because none of this would happen without what I call core stuff that won't win you a Nobel Prize, but will help other people win a Nobel Prize, whether it's how you measure stuff, whether it's about data infrastructures, collecting data. And behind the team you've seen today, there's a whole squadron of people. Pamela Linkstead, Bridget, Archie's here, Robin's here, who've actually basically dedicated their lives to this. And we mustn't forget that. This is about teams at scale. So a huge thanks to them, Caroline Hayward, but also the funders, the people in the early days. It was Roland Jung, who was the chief scientist who stuck his neck out about this. So we should all always, what's the phrase? You stand on the shoulders of giants. So great people. So we've had, I was also going to say in 2005, what was the price of a pint of beer? Blair? Oh gosh, he's clearly drank in the student union because it was £2.05. It's gone up to £6. This didn't come along until 2007. And we've now got powerful AI-assisted pocket computers. We can't imagine what's going to happen in the next 20 years. It's going to be incredible. And I do think detail and scale is important. And I remember John Saville used to say, Scotland needs to convene as a single research and care site. Because only if we get that scale and partnership will we deliver benefit to the Scottish population. And that's our opportunity. So lots of work. So we'll book you up. I know your diaries are busy, but we'll book you up for 20 years' time to see how far along this track we can go. But there's one thing that I want to say, because these events don't just happen. Firstly, can we thank the catering staff, who I think were terrific. And the beautiful surrounds of the Playfair Library. But none of this would have happened without Fiona. And Fiona. So, Fiona, if you could do the 100 meters and join us at the front, because we've got a small gift for you, just to say thank you on behalf of us all, because I think it's been a terrific celebration. Thank you. And on that note, it's been a fantastic day. Thanks for the active participation. And we'll see you in 20 years, but hopefully on many occasions in between too. So thank you very much.